Okay, good morning, everyone. Welcome to day 2 of Healthy Soils short course through Department of Viticulture and Enology. We had a very good session yesterday, so hopefully it will be as lively as yesterday today as well. Couple of announcements. If you have your questions to the panelists or through the roundtable at the end, please post them in a Q&A. And we will start in a few moments. Our announcement today is that we will have our next online meeting on winery regulations on 4th of March. Registration should be coming up shortly with that. So our first speaker today is Dr. Brad Hansen. He's a faculty member in the plant sciences at University of California, Davis. He works with ground management, mainly weed control of unwanted species in orchards and vineyards. So he has quite a bit of experience training students doing these research projects in wine grapes, as well as table grapes. So without further ado, I'd like to turn it over to Brad so he can cue up his talk and get the meeting going. So I'll turn off my camera and mute myself.
Okay, thanks, Con. Can somebody confirm that I'm on the right slide here, that you've got my title slide, please?
Yes.
Okay, perfect. I've done this a couple of times where I think we're looking at something different than we're actually looking at. So I appreciate the opportunity to visit with you all. I wish we were doing it in person. This is a lot more interesting and engaging with live people, but such is the world right now. So I want to spend the next 30 or a little bit longer minutes talking about some general ideas of weed management applicable to vineyards as well as other crops. Starting talking about biology leading into the management. For those that don't know me, I think I've actually spoken at this meeting several times, but I spend a bit more of my time working in orchards. I am the statewide extension weed science specialist for trees and vines. Largely that's been focused on tree nuts in recent years, but also working in grapes, as Con said, raisin, wine, and table grapes. And then in recent years, my group's been doing a little bit more in some annual crops, particularly tomatoes and some other annual vegetable and grain crops. But largely I'm focused on the permanent crops, and there's a lot of commonalities between trees and vines from a weed management standpoint. So just a little bit of an outline of where I'd like to go today with my presentation, and then hopefully leaving plenty of time for some questions either after my talk or in the Q&A session after the fourth speaker this morning. But I'll give you a little bit of an update on herbicide use in vineyards and a few updates on herbicide registrations, a little bit of an update on herbicide resistant weeds. I'll talk about just a few kind of new-ish weed control issues that vineyard managers and PCAs may want to be aware of. I'll talk a little bit about kind of the other kind of herbicide resistance, mostly about market factors driving some of the herbicide choices that we choose or don't choose to use. I'll give a little bit of an update on some sequential herbicide program work that we did in tree nuts in recent years, but I think is applicable to vineyards, particularly for some specific weed management issues. And then I've got a little bit of a sampler platter of some project updates that I wanted to just present a slide each to give you all a bit of an idea of what we're working on. And I'll close with kind of my just summary statements about prescriptive and thoughtful weed management to wrap this up and lead into some questions. So first of all, I'll just bring us all back to integrated weed management. This is, integrated IWM, or sometimes IPM, as an integrated pest management. It's really just thinking about using multiple strategies to resolve the problem we're trying to resolve, weeds in my case. we're trying to do this effectively, and we're trying to do it in a way that is economically and environmentally sound. And I think it's really important for us to think about both the short-term and long-term goals of an integrated weed management program. Especially, we want to be thinking about reproductive capacity. For most of our weeds, that's seed production, but also some of our perennials have vegetative propagules. So anything we can do to prevent or reduce spread of a new weed into an area, or a small patch into a large patch, is very beneficial, because we can do much more to manage a small problem than when it's a large and widespread problem. We want to take any practices we can to delay and suppress weed growth. Again, we're just trying to put our thumb on the scale in terms of the reproductive capacity of that weed species, as well as delay any impact on the crop, which is our end goal. I already mentioned seed production, but anything we can do to prevent seed production from weeds, that is super important, because I heard a paper recently discussed the seed bank, that's the weed seeds in the soil, as the memory of the system. And usually it's a memory of poor weed management earlier, but because there's so much a long viability of most of our weed seeds in soil, a failure of weed control in 1 year could lead to many years of weed control effort, because of the seed bank that could last for years or even decades in some cases. And then, to the extent possible, to reduce the weed seed bank in soil. That's not very easily done, but there are some things we can do to, in certain instances, to try to flush seeds out of the soil, or in a few cases, actually reduce viability of seed in soil. And just lastly here, any integrated weed management program is really predicated on understanding the weed and understanding the control measure. And that sounds, very basic, because it is. Not every control measure works equally well on every weed. So you need to understand what it is you're trying to control, and then what the strengths and weaknesses are of the particular strategy. Using an effective herbicide at the wrong time, or on a weed that is not sensitive to that particular herbicide, is not going to be a good weed management program. Likewise with our physical and non-chemical approaches as well. A good strategy used incorrectly is a recipe for failure. So I want to use this slide just to lead into my herbicide talk. You guys all know this, your vineyard managers, your PCAs, but one of the things I think about a lot as I transition between permanent crops and annual crops is this dichotomy of our management zones, where we have often relatively intense management right under the vine row or right under the tree row in the case of tree crops, and then a much less intensive management in all of those middles or alleys. And that's certainly beneficial in some ways and detrimental in other ways, particularly with regard to seed production. This is a bit less applicable in vineyards, but I think about the weeds that are allowed to grow in the middles of orchard crops fairly often. Again, going back to my seed comments earlier, if I have 100% weed control in my strips and 0% weed control in the middles that I allow to go to seed, I'm going to keep reintroducing weed seeds into those strips that have to be managed year over year. So it's important to think about the whole system as you're developing an integrated weed management program. I'm going to flip this slide up just as a reminder. This is a chart that I try to update every year or a couple of times a year of the tree and vine herbicide registrations. This is something I put together a few years ago, mostly as a cheat sheet for myself, just to keep track of what was registered or not registered, or perhaps has a non-bearing, that's the NBs on this label. So one of those columns is grapes, and this is broadly across all market classes of grapes. So this is something you can find online. It often shows up in newsletters and so on. So look for this if it's something that would be helpful to you. A few years ago, I put together that colored column, the second one from the left, which is the mode of action group. So if you're thinking about resistance management and trying to get a easy reference for different herbicide modes of action in terms of your resistance management programs, this might be a useful resource as well. This isn't easy to see, so I'm going to just pull out all the grape information to make a few comments here about the herbicides that are currently registered for use in grape in California. And that's, again, across all market classes. So the two columns on the left are our pre-emergent herbicides. These are the ones that we apply to, preferably to bare soil, before the weeds germinate and emerge. And the two right-hand column and that lower organic column on the lower right are post-emergent materials. So I'm not going to go through each of these, but I do want to make a couple of comments here about some key herbicides for use in grape. As I move through here, a couple of comments, particularly on, I would say Allion has been one of the herbicides that's been really important in recent years from a pre-emergent standpoint. I see quite a lot of that as sort of a foundation material, as well as Chateau and the other products with that same active ingredient as Chateau. Mission is one I've seen increasing a bit in grapes, kind of filling the same slot as Matrix. It's in the same mechanism of action group. Comments here, Surflan is an old standard that has been kind of decreasing in use to a degree. There's probably some market chain issues with Surflan, so that's one that's been a bit less well supported, and I think that trend may continue. One comment I would make here about Zeus, this has been registered for a number of years in grape, but has not been very readily available in grape because of, again, I think that there was a time-limited registration. I did speak to a manufacturer person a couple of days ago, and they're actually expecting to have Zeus back available in California by the end of 2021. So too late for this winter, but probably in time for next winter's application. So that'll be a bit of a one more tool for use in grape. Comments on the post-emergent materials. Let's see here. Probably the big, well, I'm going to come back to glyphosate. Everybody knows glyphosate, but I'm going to talk about herbicide resistance here in a minute. I'm going to talk about some of the market factors. So I'll leave that one aside for the moment. A few of these grass herbicides, particularly Select Max and Post, and Fusilade, and the other generics in those mechanisms of action are quite widely used for grasses. Select Max has a non-bearing label in grape. Interestingly, though, if you work in tree crops, there's been some changes with the Select Max and the other clethodim herbicides where they can be used in bearing tree nuts at this point. So I'm curious to see whether we see changes in grape as well eventually. I see some comments in the Q&A about stop orders for weed slayer. I will come back to that in a few minutes. So other movement in this post-emergent group of herbicides is the Rely 280 and the similar herbicides. So Rely 280 went off patent a number of years ago, 3 or 4 years ago, maybe 5. And there's a whole bunch of herbicides with that active ingredient. So that's been very widely used because the price has been coming down a bit. It also works, it's not quite the same as glyphosate in terms of its efficacy, but it's a fairly broad spectrum herbicide. So that active ingredient has been fairly widely used. Let's see here. Question. While I'm here, somebody asked if there's an organic pre-emergent. There is no effective organic pre-emergent materials that I'm aware of at this point. Okay, so let me back up one here. So this is all the herbicides that are registered in grape. We don't use them equally. So just make a quick comment here. This is DPR, pesticide use data for all grape classes. And I've got here 2014 and 2017. I think when I updated this a few weeks ago, the 2018 data were not quite out, but they should be available pretty soon. Just a quick reminder here as well, that these herbicide use data are sprayed acres. That's not the same as vineyard acres. So remember that strip applications that I discussed. So if it's a 50% wide strip, that ends up being half a treated acre, right? So just to put that in context here, so the most widely used herbicide in grape still is glyphosate, and has been, since I've been doing this, very, very widely used. So there's about 500,000 acres, sprayed acres of grape treated with glyphosate. That's out of, what is that, 900,000 acres, vineyard acres of total grape acres. So that's roughly once a year, maybe a little bit more than once a year on average. Interesting trend for glyphosate. This is actually starting to come down. These brown arrows on the right are relative to 2015, but you can see the trend, slight decrease here. I'll come back to this, but I think what you're seeing with glyphosate is twofold, probably partially related to glyphosate-resistant weeds, but almost certainly driven more by market factors because of consumer concerns about glyphosate and pressure from the wineries and so on. So we are seeing a decrease in glyphosate use over time. I mentioned a moment ago the glufosinate or the Rely type products. Those are increasing, largely displacing the glyphosate acres. That would be my quick summary of that. Also, the price has been changing a bit, so it's become a little bit more economical. Interesting downward trend for oxyfluorphane. This is the Goal and Goaltender, and there's a Galagan, and there's a whole bunch of oxy products out there. Seeing a slight decrease over time with those, but still a very important set of herbicides. Let me see. I'm not going to go. I don't think I'll go through all of these. You can find your favorites here, but I'll just pull out a few more highlights. Seeing a bit of a decrease versus the previous year in Chateau and the flumioxazin products. Kind of static in terms of the pendimethalin, which that surprised me a little bit because typically the pendimethalin would be displacing the oryzolin. That's a similar type of herbicide. Let's see here. And I mentioned the increasing flazosulfuron. Mission was registered probably in about 2012 or so, and has become fairly important in vineyards, kind of filling the same role in a tank mix as the Matrix. So there's a bit of head-to-head pressure, I would say, with Matrix and Mission, or Rim Sulfuron. There's a lot of Rim products out there. Matrix is kind of the flagship. Okay. I see a few questions coming in. I will try to, if I catch them as I'm talking, I'll try to answer them. Some of those I'll wait and try to address at the end as well. Okay, quick update on glyphosate resistance. This has been the driver for my research program for most of the last 10 years because glyphosate resistance kind of became really important, although I think we're doing a fairly good job of managing it at this point in tree crops in most tree crops, as well as in the vineyard system. So you can see the list of the rogues gallery here of glyphosate resistance in California, orchards and vineyards, I should say. I haven't added any new weeds to this list in a while. In terms of broadleaves, I would say statewide, probably hairy fleabane is our biggest, most widespread problem in the Central Valley vineyards and orchards. In some areas, horseweed edges out fleabane. Those are related species. Like if you get in the Fresno area, probably horseweed is more important, but statewide, I think fleabane is a bigger problem. The Palmer amaranth, that's one we actually have seen some cases of resistance in vineyards, a few in orchards, and then more in some glyphosate resistant annual crops like cotton and corn. But it's certainly around. This is a hot weather loving plant. We tend to see it more in the San Joaquin Valley, less so in the coastal production areas because it's just not that well suited. In terms of the grasses, again, no new ones added here. Ryegrass is probably one of the major problems in the North Coast and the Sacramento Valley in terms of our perennial crop production systems. We've had some reports and confirmation of annual bluegrass. That doesn't tend to be a major vineyard problem. We see it more in the orchard systems. And then I've had quite a few people in my lab work on jungle rice in recent years. That's a summer annual grass. That's one we've seen in some San Joaquin Valley vineyards, but it tends also to be more of an orchard problem. Okay, and then I just gloss over, I guess, here. there's a group of summer grasses that I'm getting more questions about, although we haven't really confirmed resistance. But three-spike goosegrass, that's a major tree nut problem because it's a perennial grass. Windmill grass, sprangletop, witchgrass, these tend to be more San Joaquin Valley, hot weather, hot climate loving plants. So we're getting more questions about glyphosate resistance. And these all make sense because they're in these genera that have glyphosate resistance around the world. These are the Illucines, the Chloris, Leptocloa, and others. So it's very likely we have broader resistance as well. One of the bigger challenges in recent years with resistance, I mentioned not really reporting a lot of new glyphosate resistance problems. What we have done in the last 6 or 8 years is worked on a couple of key species that are resistant to more than one herbicide. So these are primarily already resistant to glyphosate and then evolve a further resistance to something else. In the two photos on the right there, the almond orchard on the top has glyphosate and paraquat resistant hairy fleabane. You can see the fleabane in the strips. I think that orchard had been treated 3 or 4 times by the time we took this photo. Likewise, that prune orchard in the lower right has glyphosate and paraquat resistant annual or Italian ryegrass. Similarly, it had been treated several times when my former student took this photo. So the stacked resistance is becoming, I think, a bit more problematic. We've worked on the paraquat stacked resistance mostly, but there are cases of resistance to paraquat, the ACCase herbicides. That's the things like Fusilade and Select. That'll be in ryegrass. And then a few cases of resistance to glufosinate, the Rely type herbicides. So, be very cognizant. If you see failures of control, don't brush those off because it's very possible you could have a new case or a beginning problem with a multiple resistant weed species. All right, I just want to hit on a few of these species specifically quickly. I'm not going to belabor this too much, but the ryegrass complex, that's a couple of different Lolium species, mostly Italian ryegrass, but also rigid ryegrass. This is a species that's very, very prone to develop resistance around the world. There's populations, particularly in Australia, that basically can be controlled with almost any herbicide mode of action. Resistance to glyphosate is very, very widespread in California, and it's probably the rule more than the exception. I just mentioned we've had reports of multiple resistance to also from glyphosate resistance and resistant to paraquat, some glufosinate, some of the ACCase herbicides, and a few of the ALS inhibitors. That actually is herbicides like Matrix and Mission. I have not really seen problems with ALS inhibiting herbicides in vineyards, but it can be a major, major problem in some of the cereal grains that have a much more heavy reliance on the ALS inhibiting herbicides. Okay, the two Caniza species, I mentioned hairy fleabane and horseweed, so just the photos there so you can see the difference there. As I said, I think fleabane is a much wider spread problem in the state, although there are pockets where the horseweed is the major issue. I think part of this is because fleabane has a shorter life cycle. It has a much faster turnaround in terms of its seed production. Like fleabane on the left photo there, it'll be making seed by probably June in a lot of the Central Valley, whereas the horseweed is usually several months later, more like August and even into September. So let's see here. But similar to ryegrass, glyphosate resistance is really the rule more than the exception with the two Caniza species. This is actually a very interesting resistance case because there's really an age and size component to this as well. If you have a very good timely application, very small fleabane and horseweed might be controlled with glyphosate. I'm talking the size, the diameter of a quarter or something like that, very small. Conversely, if you let those plants start to bolt, where they transfer from a rosette stage to growing taller, almost no population is completely susceptible to glyphosate at that point, even if it's quote unquote susceptible. as once they reach that bolting stage, they're very, very difficult to manage with glyphosate. So this is where that integrated management program I discussed is important. Getting out there with a timely application will certainly give you a better chance of success than a poor timing. Okay, this is sharp point fluvelin. This is actually a slide I lifted from one of John Roncaroni's presentations, but this is some of the species I talked about previously are really widespread problems in orchards and vineyards. Sharp point fluvelin is one I tend to see mostly in vineyards or non-crop areas. This is an interesting weed for a couple of reasons. I mean, first of all, it's an annual, so it produces by seed. It's interesting because it germinates pretty late in the spring or even in the summer. So oftentimes we see this one coming late. We might have had really good weed control in the winter time and controlled everything else, and we've left an open niche. And then this species that's able to germinate during the warmer periods of the year is able to come on late and exploit that open niche. Seeds are pretty long-lived. One of the interesting things about sharp point fluvelin is the juvenile plant, the one you can see in the middle of the photo there, looks quite different in terms of its leaf orientation than the more mature plants. So sometimes this weed is misidentified because the leaves are opposite one another early, and then later on they become alternate. So sometimes it can be a challenge to identify. This can be it's fairly difficult to wet. It's a very hairy species, so it can be difficult to wet with contact herbicides. It's actually not that hard to control. Some of the herbicides like glyphosate work fairly well on it. It's just that it tends to come on so late that we are not able to use that herbicide in the vineyard because of concerns about crop safety after the canes start to elongate. So this is one where we do see problems late in the season, even following effective control of our winter species. Field bindweed, I think most of us who work in agriculture are fairly familiar with this species. This is a very difficult weed for a number of reasons. One, it can reproduce by seed as well as by vegetative propagules. It spreads through creeping roots and rhizomes, and you can see in that middle photo some of the examples of really long root systems. Because of that deep and extensive root system, it's very, very drought tolerant. So you'll see, like in that photo on the left, I think that's a fallow field adjacent to a tomato field here on campus. Incidentally, on campus, I think my soil test has sand, silt, clay, and field bindweed. We have very, very dense populations here. But it's an extremely difficult weed to control. And because it's a perennial, it's really difficult to manage with our pre-emergent herbicides. We can do an okay job on the seedlings, but those perennial structures typically can grow through our pre-emergent materials. Because it's a perennial, it doesn't respond very well to our contact herbicides. You'll burn it off with a lot of things, but it tends to regrow. It's very, very, it's got a lot of reserves. Glyphosate tends to be our best option here because that's a translocated herbicide. So as we start moving away from use of effective translocated herbicides, I think weeds like field bindweed are going to be an even more important issue. I actually had a call recently about a biocontrol agent for field bindweed. There is a mite that's been released in some other states that's not been deregulated for use in California. I'm a little bit hopeful that the biocontrol program may pick this up, but I would just make the comment here for anybody who's hoping for a biocontrol solution. It'll be pretty slow and pretty, biocontrol in ag tends to be kind of suppressive rather than an eradication level of control. But again, going back to my integrated weed management comment early on, if we can do something to, disfavor field bindweed, I think that's to our benefit. Another species this is another one I tend to see mostly in vineyards. The willow herb, we have two willow herbs, the two epilobium species, fairly similar in terms of kind of the niche they fill in the vineyards. These are annuals. They're windblown seeds, so we see these very frequently after disturbance, so after a tillage operation or a new planting. Other epilobiums are commonly called fireweeds. We see them in natural areas after fires because they're able to blow in, and seeds blow in and they kind of recolonize their early succession species. One of the reasons they're problematic in agriculture is that they're fairly tolerant to glyphosate. In vineyards, that's our most widely used herbicide. Glyphosate tends to stunt and kind of delay willow herb for a few weeks. It'll turn a little bit red, it'll stop growing, and then it'll start growing again a few weeks later. So for me, as I drive around the grape production areas, I feel like I can pick out most of the glyphosate-only vineyards because they've got a good crop of willow herb, and perhaps they've had fairly good control of other species. Bermuda grass is another problem. This to me is kind of in the same category as field bindweed. These really densely growing perennial plants are going to be very big problems as we have fewer translocated herbicides. Like field bindweed, this species reproduces by seed as well as by stolons or rhizomes. Stolons are below ground creeping roots, or below ground stems, and rhizomes, sorry, the other way around. Rhizomes are below ground, stolons are above ground. So we can see this creeping both below and above ground. Very, very drought tolerant. This is a summer growing species, so it'll typically go dormant in the winter, and it'll start greening up probably here pretty soon as the soil temperature warms. Different though than field bindweed, we do have some other herbicides that are grass specific that work fairly well on perennial grasses. The Select, Fusilade, Prism. There's several of those ACC-ACE inhibiting herbicides that work fairly well on grasses. So we've got a few more options here with this species than we do with things like field bindweed. Okay, just a handful of more weed species I want to touch on. The Malva, there's several Malvas that we have. Mostly we tend to see Parviflora, which is cheeseweed, but we do have some areas where Malva neglecta is common. This is it's an annual species technically, although we do sometimes see individual plants that overwinter. I call those yearlings. They get really woody and they can persist, particularly if it's a mild winter. These reproduce by seed. These seeds are really hard and really durable, so they can last very long time in the soil. And actually, in a previous position I held, I did soil fumigation work. And this is one of those species that's even difficult to kill with fumigants because the seed coat is so hard and protective of the embryo. Like some of the others I talked about, this is a problem because it's very difficult to control with our key herbicides like glyphosate. So one of the reasons we see a lot of the oxyfluorphan products used in our ag systems is because the oxy herbicides can really help a lot with Malva and a couple of other key species that glyphosate is a little bit weak on. So this is a species that's still germinating now. We tend to see it in the late fall into the early winter. And in this middle photo, you can see the cotyledons. So this is a very young plant. You can see just the first couple of true leaves, and that heart-shaped cotyledon is one to watch for if you're seeing seedlings come out. Very likely that's one of the Malva species. Okay, nut sedge. This is one, the nut sedges, I should say, we mostly have yellow nut sedge, but in the desert production areas, there is areas where purple nut sedge is the bigger problem. So some people call this nut grass. I just want to make a comment here that it's not a grass, it's a sedge. That's a different kind of class of plants. It's a perennial, but it's an herbaceous perennial that typically dies back. This spreads mostly by vegetative propagules, the little tubers, the little nutlets that you can see in the lower right photo. On yellow nut sedge, they grow on the end of the roots. On purple nut sedge, they'll have chains where there'll be a nutlet and then more root and another nutlet and more root. We fairly commonly see yellow nut sedge flowering. That's what this photo in the upper right is, but we rarely see seed production in California. So this is really mostly a problem with spreading tubers through tillage and soil movement in the vineyard. Okay, a few more weed species here. Jungle rice, I mentioned, we've done quite a bit of work on this. This is a relative of barnyard grass. There's also a lot of Echinocloa species that are problematic in rice production systems. This is not one I see a lot in vineyards. It tends to be mostly an orchard problem, or Roundup Ready corn and some of the other annual crops. We do have a lot of glyphosate resistance around California. And so this is one just to be aware of, although, as I said, I tend to see this mostly in the San Joaquin Valley, less so in the coastal grape production areas. Okay. So Palmer amaranth, this is a pigweed. We have lots of pigweeds in agriculture. This is one that's has been in the news a lot in say the last 15 years because of issues in other parts of the country. This is one I've seen, there's some areas. This photo is one we took in Tulare County in a raisin vineyard production system. Again, mostly a San Joaquin and hot climate type of weed. We tend not to see it in the coastal areas, or at least I haven't seen it so far. But watch out if you see a pigweed that you're having difficulty with. It may actually be one of these more problematic weeds like Palmer amaranth or common water hemp. Okay. And then I mentioned this group of summer grasses already, so I'm not going to belabor this, but there are, besides jungle rice, there are several other summer growing grasses that are becoming a bit more problematic depending where you are in the state. Okay, switch gears, one more weed slide, sorry. So as vineyards expand into more areas, I'll get unusual early vineyard or early orchard questions about weeds that I would classify as kind of rangeland weeds. I get more of these in vineyards because you guys grow vines on some of the more rolling, difficult topography versus say almonds or walnuts. But we do see some of these new species. I would say the one we've done the most work on, relevant to grapes, is probably perennial pepperweed. If you get in the Sierra foothills, there are some areas where perennial pepperweed. A few cases of rush skeleton weed, which I don't have on here, but there are some difficult. These are perennial weeds that are difficult to control. Some of these others, like alkali mallow, alkali weed, I tend to see mostly as pistachio weeds because of some of the challenging soil conditions that pistachios will tolerate. But if you're in a new production vineyard, you may see some really unusual weeds, and it might challenge your weed ID a bit. Okay, so I want to switch gears, and I think this will get at a few of the questions that have come in. But I do want to talk just a little bit about the other kind of herbicide resistance. That's the market pressures on herbicides in general, or a few specific active ingredients, primarily glyphosate. So the driver for the glyphosate questions really was a 2015 designation by the International Agency for Research on Cancer put glyphosate in the class 2B probable carcinogen category. This is I've actually probably presented this slide to you all before, but this was a slightly different analysis of existing data using a hazard assessment, which is a bit different than what the regulatory agencies like the US EPA, the European Food Safety Administration, and all of the regulatory agencies that use a risk assessment approach. So risk assessment takes into account the hazard plus potential exposure, whereas the former really only doesn't take into potential exposure as part of the analysis. But there was a lot of litigation. There still is litigation ongoing related to glyphosate alleged causing of cancer, specifically non-Hodgkin's lymphoma. So there's been a lot of this has been going on. A lot of discussion and concern about this in the public sector. The US EPA in January, a year ago, released another registration eligibility review. They go through all the pesticides regularly, and they did another one of glyphosate, and came up again that according to US EPA, it's not a carcinogen. So there's still some controversy swirling around here, but interesting for us, glyphosate has been added to the California Prop 65 list because the IARC is one of the sources that puts products on, allows products to be added to the Prop 65 list. However, US EPA is the body that decides on regulation and labels, and they have made no indication, actually very forcefully indicated that it is not a carcinogen, and actually pushed back on some California regulations relative to the cancer labeling question. So I don't see a lot of impact on agriculture from a registration standpoint, from a regulatory standpoint. However, the market sector has a tremendous leverage on products, especially things like wine grapes that are very close to the consumer. So I think we will see, and we are seeing, some changes already in the use trends of glyphosate because of this market pressure. So be certainly aware of what your market wants, and take that into account. Because of that, I've given a bunch of presentations in recent years, to grape growers, to walnut growers, and so forth. Sometimes it's a little bit of the sky is falling. Can we manage weeds without glyphosate? And I would say yes, we certainly can. Glyphosate is a really useful herbicide, particularly because it's inexpensive and controls a lot of kinds of weeds. It's very broad spectrum, doesn't have any soil activity. It's translocated, so it works on perennial plants and large plants. But because of resistance, it's not really been a standalone program for a number of years. Grape growers and some of the bigger tree nut industries have quite a few herbicides. I showed you that registration slide early on. I think we can develop pretty good, very effective weed control programs from these options in most cases. There's not a direct drop-in replacement that does everything that glyphosate does, so it's going to take a bit more effective management and good PCA work. And costs are likely going to be more. We don't have, glyphosate is very inexpensive because it's been off patent for 20 years or so. So we're going to see some increase in cost using other herbicide programs. But I think we certainly can manage weeds without glyphosate, whether, if we choose not to use it or if we're disallowed to use it for whatever reason. So what are the challenges going to be? I said, well, first, the good news is you don't have to worry about glyphosate resistant weeds anymore. So you can worry about all the weeds because they're the same level of problem at that point. The biggest issues are going to be the first two bullets there: timing. Large weeds are difficult to control with our contact herbicides. Glyphosate is fairly forgiving of coverage and of size of weed, assuming they're susceptible. So we're going to have to be better about timing in terms of getting a contact herbicide or a pre-emergent herbicide on in timely fashion relative to the weeds phenology. Those perennial weeds, particularly things like yellow nutsedge and field bindweed, are going to be probably a much more important problem because we just don't have very many herbicides that work well on established perennials with extensive root systems. Timing could be an issue, timing of the pre-emergent materials if we rely more on those, timing of the post-emergent materials relative to size, and then the economics are going to be a big factor here because I don't think anything is quite as cheap as glyphosate. So it's gonna have a cost increase to a degree. I need to speed up here a little bit. I realize I'm kind of going a little bit slow, but I want to hit just a couple of points about some sequential work. This was actually work we were looking at summer grass control in tree crops, but I think some of the lessons are applicable to grapes as well. So this was a walnut orchard site. And you can see the seed on the photo on the right. That's the kind of jungle rice density we were looking at in that orchard, glyphosate-resistant jungle rice. What I started thinking about with this is our winter treatments. What we typically do is we put our pre-emergent herbicides on our strips in the grapes or in the trees in December or January, and then that lasts for some period of months before it starts to break. Usually that's going to be kind of just leading into our summertime weed germination. So that could be things like jungle rice in this case, or in the vineyards, it could be something like sharp point fluvelin. So the ways that we try to resolve that, we can use a higher initial amount of herbicide so it lasts longer. We use the top of the label rate instead of the middle of the rate to try to get it to last longer. That always seemed like kind of weird approach to me. So what we wanted to do, what I had Kaio do to demonstrate, was basically a follow-up application. In this case, we were using an early April 2nd application of a pre-emergent herbicide, trying to stretch weed control into the summer months. And just a quick rundown of this, I've got just this one data slide. But basically what I wanted to do, we had 3 different foundation programs. Allion, which is registered in trees and vines, Pindar GT, which is registered in trees but not vines, and then Tuscany, which is a chateau type product. It's flumioxazin. So each of these we had a base program, a foundation program, and then we had a base program plus prowl in the winter. That's 4 quarts of prowl. So let's use a grass herbicide to try to give us longer control of grasses. We had a followed by program. The FB is followed by. So that was a winter base program followed by a low rate of prowl in the spring, early April in this case. We had a followed by the high rate, and then we had a 2+2. So we had a 2-quart rate in the winter with the foundation program, followed by a 2-quart rate in the spring. And you can see here, anytime we added, well, in each of these cases, the followed by program gave us much better weed control. Long bars are better weed control in this case. So this is an August rating. So this is a late summer rating. And what I took home from this is by thinking about the weed biology, that this is a summer germinating grass, and thinking about our herbicides, when can we get that pre-emergent herbicide on before this summer grass is germinating? We had as good of control with half as much herbicide. In this case, 4 quarts of the herbicide used at a less effective time of the year was not as good as half as much herbicide as a more appropriate time of the year. So this is a slide I like to use to make that point. We want to understand the weed biology, and we want to understand the herbicide or the tools. This applies also to our non-chemical approaches, but using those tools to their best ability will give us more effective weed control and hopefully at a more economically and environmentally sustainable fashion. Okay, my last couple of minutes, I want to hit on my sampler here. So we'll be like this guy. I'll try to give you a sample of everything in just a couple of minutes. So this is one that's sort of unique to grapes, I think. I get this question a bit: what age is a new planting? Particularly as we start thinking about pot dormant or dormant green vines and some of the pre-sleeved young vines. A lot of our herbicide labels say a year old. And I just want to make the point here, if you're considering crop safety, err on the cautious side here. A vine that's been in a pot for a year and been in the ground for a month is not a year-old vine with regard to herbicide safety. So some of the labels are a little bit squishy on what that means, and because I think a lot of them were written prior to some of this new technology that we're using in grapes, as well as in some of the non-dormant plantings of tree crops. But I think just it behooves us to be cautious about how we use herbicides with regard to young plantings. I think it's also really important to have sleeves on our cartons on our vines early on too, because that does give us an extra degree of safety from our contact herbicides. One of my messages I like to share is most of the herbicides that we use in trees and vines can injure trees and vines. So we try to keep them above the root zone and below the leaves and off the stems. That's how we get most of our safety. So anything you can do to be cautious here, I think is important. We get away with things a lot, but we don't get away with them all the time. So, just be cautious if you're working with young vines. A quick comment here. This was not a grape study, but about in 2008 or 10, there was a lot of controversy, a previous controversy about glyphosate. At that point, it was discussion about being a micronutrient chelator. I think a lot of micronutrients were sold into the tree and vine system because of this, but it never quite made sense to me. So we conducted a study, a 6-7 year study. I planted a block with almonds, prunes, and cherries. Half of those were amended with a very sandy soil. For a couple of years, we put flood irrigation on right after a glyphosate application. So the worst case scenario here, we had three applications per year a 4x rate, so about a gallon per acre of Roundup Power Max, 3 times per year, 6 years in a row. So the worst case scenario had 18 applications of a gallon per acre over the course of that experiment, 72 pounds of active ingredient. And then we looked at micronutrients in the leaves, and we looked at a bunch of growth parameters of all those trees, and really had no measurable impact. So to me, this says the micronutrient deficiency question is probably not very likely to occur. And we didn't really see any growth effects that could be attributed to glyphosate at all. As an aside, I would say that much glyphosate will actually control field bindweed fairly well, but it's not a recommended practice either. Couple, as I wind down here, this is obviously an orchard focused slide, but one of the things I've been thinking about a lot is this herbicide intensity. And particularly in like that right-hand photo, you can see a very, very intense herbicide program in almonds. And I've been thinking a lot about how can we manage weeds where we need to manage them, but use less intense programs in areas where we can, and what we can do from an environmental sustainability standpoint. And I think some of those lessons are applicable to grapes as well. Where we can, I want to use the tools well, but not overuse the tools. And I think anything we can do to, narrow up our spray strips or use a pre-emergent herbicide so we don't have to use as much post-emergent herbicide. These are all ways that we can collectively move the needle in this more sustainable direction. In a few cases, I've been working a little bit on some chemigation approaches, trying to, again, think about can we use the herbicides only in an area where we need it? And this may not be as applicable in vines, although we've done some drip chemigation as well. That got shelved last year because of COVID-19, but we'll pick that up again in the future. In orchard systems, we're doing some cover crop work. This is already fairly widely used in vineyards, but this is something that we're, again, thinking about system-wide sustainability. Okay, somebody asked a question early on about the Agrigold weed slayer stop order. So that was pretty big news in late December. There was this co-packaged product with a organic fertilizer and the organic herbicide weed slayer was contaminated with some non-organic pesticides. So as far as I know, this is currently off the market while they sort that out. So I assume if you've used this, you've heard about this and are no longer using Agrigold weed slayer, Agrigold WS rather. So I guess I don't know much detail on that, but this is certainly one to be aware of. This is really unfortunate because there was a lot of really good reports of quite a bit of efficacy with this herbicide, this organic herbicide. So I'm hopeful that that's a small scale problem and we'll see it back on the market, but I don't have any information about that. Okay, let me see. Just a few regulatory comments. This continues to be a challenging part of our industry. There's a number of our pesticides have some tox and worker safety issues that's changing handling systems. This is largely related to paraquat, although it's been proposed for some other herbicides as well. Some export market concerns with residues. I saw there was a question in the chat about MRLs. So this to my knowledge, this hasn't actually changed anything, but there's a lot of discussion in the European Union in particular about changing MRLs and what a disharmonious MRL. If the EU has one MRL and the US or other markets have a different MRL, what that means for growers. So there's a lot of concern about what that means for us in grapes as well as in other tree crops. Lots of consumer and buyers leveraging changes to the production system. That's especially true of things like glyphosate, but even more so the sustainability and healthy soils goals. So we're seeing a lot of kind of non-regulatory pressure that's changing how we do things. And the bad news, I guess, I don't see this getting any easier for the growers. So just to wrap up here, all of our weed management choices have consequences, including doing nothing. I spend a lot of my time talking about herbicides, but herbicides are only one tool. but we should use all of the tools as part of our integrated management approach. And whatever tools you use, whether they're chemical or other, use them to their best ability based on an understanding of the biology and of the tool. Okay, and lastly, don't just use a bigger hammer to try to get the job done. Use the right tool for the job. And I'm going to stop there. I see Khan showed up. That probably means my time's about out. But I will be back for the Q&A, and I'll try to address questions in the chat while the next speaker is speaking. So thank you very much for your time.
Thank you very much, Brad. Unfortunately, we're out of time. I want to bring on Christina Lascano back. So as Brad is attending to the questions in Q&A, I'd like to reintroduce Christina Lascano from Land, Air, and Water Resources. She's not so new, but she's relatively new to UC Davis. So as I alluded to yesterday, we put together a very nice group at Napa County to study the effects of soil health, or rather survey the health of soils. And Christina is leading this project, so she will give us a little update about it with the grander scheme of things with soil health. So I'll turn off my camera so you guys can see Christina better.
Hello, everyone. So I hope you can see my screen now. All right. So hello again. Welcome back. And in this presentation, I'm going to be focusing on how we can assess the health of vineyard soils. And so if you were here yesterday, you remember that we introduced the concept of soil health generally and the properties of a healthy soil. And then we moved into management strategies that we can use to improve soil health, how they increase soil carbon, and any agronomic and environmental benefits as well of those practices. Now, in this presentation, I'm going to revisit the concept of soil health, and I'm going to evaluate it critically and with the perspective of wine grapes. So what I'm going to do here is actually show more of the research gaps that are out there and the research that we are currently working on. And so you probably have a lot of input and questions, so please put your questions and any comments in the chat, and I'll try to address them later. So we define a healthy soil as a soil that is able to function and support the life of plants, animals, and humans. And there is a series of agronomic, chemical, physical, biological, and environmental properties that are typically characteristics of a healthy soil. And we learned that yesterday. So to assess the health of a soil, what we do is to look for measurable properties that are related to these functions. So that's what we call soil health indicators. And so these are, again, things that we can quantify either in the field directly or in the lab through different types of analysis. For example, if we want to assess the health of a soil in terms of yield, right? We need to look at what's the harvested crop per area. If we want to look at the quality of the crop, this kind of looks different depending on the crop. So for grapes, we want to look at sugars, phenolics, acidity. Other crops, maybe it's more interesting to look at vitamins, micronutrients, etc. If we want to assess biological properties, we can use different techniques to assess diversity, like genetic markers, and I'll explain more about this later. To assess activity of the microbes, we use different tests, but one of the most common is respiration, which assesses the amount of CO2 that is emitted by the microbial community. But there are other options as well, and I'll talk about this later again. If we want to measure the environmental efficiency or outcomes of the healthy soil, so we can measure, for example, how much carbon do we have in the soil, and we can do that in percentage, in concentration, etc. And there's also different types of carbon that we can target, right? Different like active carbon, total carbon. In terms of like how much the soil reduces emissions, we can assess the release of CO2, nitrous oxide, or methane from the soil. If we want to assess the capacity of soil to supply nutrients or also to recycle nutrients, what we look at is like the amount of available nutrients in the soil at a certain moment, but also the potential for those nutrients to be released and recycled, like mineralization rates and nitrification rates processes, right? We talk about soil health being a dynamic concept. And then in terms of soil physical properties, we are very interested in soils holding on to water, but also infiltrating and not getting too water saturated. So we looked at things like water holding capacity or infiltration rates, and infiltration rates can actually be measured in the soil very easily in the field. So this is an example of soil health indicators that are commonly used, but it's not a comprehensive list. Now, something I would like to mention, and this is something that it's actually being discussed a lot by the scientific community right now, is that the properties and indicators of soil health that are targeted through management are usually way differently for different crops, also depending on production goals and specific site challenges. And what that means is that the meaning and the definition of soil health can actually be quite different for different crops, like for wine grapes or corn, right? Both crops have different requirements. They are grown generally in different areas. And so what we are looking for on a soil, what we need of a soil, it's also different. And unfortunately, most of the research that we have so far on soil health has been focused on corn and soybeans, and in areas where these crops are grown that are extremely different from California. And I'm going to illustrate a little bit these differences and why it's important to generate a definition of soil health that it's crop specific. So again, corn and wine grapes are typically grown in very different regions. So corn on the one hand is mostly grown in the Midwest, and then grapes are mostly grown in the west of the US, as an example, more typically Mediterranean arid areas, right? There's there's different types of soils in the two regions, which is very important. And the needs of the crops are different. So for instance, the nutrient uptake of corn is very high. So the nutrient demand is high. The one we want of a soil is to be able to supply, to produce high yield. That's also the focus of producers. In wine grapes, on the other hand, we're interested in yields, but we are also interested in quality, right? That we want a specific chemical profile on the grapes that it's going to be associated also with a later quality in the wine, right? So to be able to produce these high yields, as I mentioned, because corn has this very high nutrient demand, what we want in the soil, it's a high nutrient supply, but also if we are environmentally conscious, we don't want to supply too many nutrients that can be lost through leaching or through nitrous oxide emissions. So we want a soil that retains the nutrients and recycles them internally, right? In grapes, we want a soil that it's able to supply just the right amount of nutrients. We don't want a lot of nutrients. And what we're interested as well is in the soil that recycles and retains nutrients. In California, because of the issues that we have with droughts, we're also interested in water. So we want a soil that it's able to hold on to water, right? As long as possible. So we want a soil that stores water. So we need a definition that it's crop specific, that it's tailored to the needs and particular aspects of wine grapes. Now, the importance of different types of soils and soil properties for wine grape production, it's not a new idea. And this is something that has been already incorporated into the concept of terroir. And so the wine industry knows very well the role of soil in producing high quality grapes, right? And so the terroir concept incorporates the impact of soil when combined with the climate, the grape variety, the topography into the quality of the wine. But the aspects of soil contained into the concept of terroir are quite different of the aspects of soil that are actually considered in the definition of soil health. So even though these two concepts overlap somehow, there's properties of soil health that haven't been considered so far traditionally in the typical traditional concept of terroir. And so this is what I'm illustrated in this chart here, right? When we talk about terroir, the soil properties that have been studied the most are physical properties like depth, texture of the soil. We're talking about infiltration, structure, water holding capacity. Also, chemical properties like the supply of nitrogen, cation exchange capacity, pH. Those are properties that are very well known, and how they affect the, the quality of grapes and the quality of wines. Now, the concept of soil health slightly overlaps with the concept of terroir, and so things like soil structure, infiltration, water holding capacity are also contained into the properties of a healthy soil. But again, there's new varieties, new soil properties, like for example, soil carbon, soil organic carbon, and biological aspects of soils, like the diversity and activity of the soil microbial community that haven't been considered so far. So managing for soil health might actually be allowing for a full expression of the terroir and promoting the quality of the wine grapes. But we need to understand what's the role of these other varieties that we haven't considered so far, and the role in wine grape production, right? And so we might have to expand this concept of terroir to incorporate these varieties, right? And in particular, I want to focus on the biological component of soils, specifically the microbiome, because the microbiome regulates very important ecological processes in soils. It regulates the turnover of nutrients. It regulates decomposition of organic matter and carbon sequestration. Those are important for fertility and health of the soil. But also microbes are important for wine fermentation, right? So the yeast in the grape actually can come from the soil. So it's important when we're starting to think about managing soil carbon and soil organic matter, how are we shifting the soil microbial communities, and how is this going to be translated into the grapes and into the wine, right? If we want to study the soil microbiome, then there's different things that we can look at. The general definition of microbiome includes all of the microorganisms that can exist in the soil, and that is viruses, archaea, bacteria, fungi, protists, and sometimes other types of microorganisms, eukaryotes like algae as well, right? We can find algae in the soil, microscopic algae. Now, probably what you heard of and what's normally analyzed in most of the soil tests, it's only bacteria, archaea, and fungi. The rest are, we still don't understand very well what the role is in all of these functions in nutrient cycling and carbon sequestration. We are starting to do some research though on these other groups, and it's specifically interesting the results on soil viruses. But we don't have enough information so far. Now, when we want to study the soil microbiome, what do we need to look for? So there's different things we can analyze. And that's usually divided into two types of variables. We talk about the structure of the microbiome, and that means who is there? And then we have the function. What are they doing, right? And so depending on what we focus, there's different variables that we can, different techniques that also provide different amounts of information, and they have different costs. So in terms of the structure of the microbiome and figuring out who is there, we have a range of analyses that are commonly used in soil and in soil testing and in soil research that range from very simple analysis and relatively inexpensive to more complex and more expensive. An example of those simple analysis is microbial biomass carbon. And it's kind of like a very rough estimate of how much microbial biomass we have there. And for that, what we do is measure the carbon that it's contained in the microbes. And again, this is a very rough measure, but it's very robust and has been used in soil science for a long time, and we still use it now. Now, a bit more complicated than that, we have PLFAs, and you probably heard about that. PLFA stands for phospholipid fatty acids, and these are biomarkers of the cell walls and the membrane lipids in microorganisms. So this technique kind of goes a bit beyond microbial biomass and breaks down the microbial community into big groups. So we get, for example, the abundance of gram-positive bacteria, gram-negative bacteria, fungi, and also different types of fungi like mycorrhizae, right? So there is a bit more resolution, a bit more information being generated, and it's also a bit more expensive. Now, moving into like the more expensive analysis, we have DNA-based techniques like 16S RNA and ITS, and those are markers of either prokaryotes like 16S, and that includes bacteria and archaea, or in the case of ITS, those are markers for fungi in the soil. Now, these are really interesting techniques that provide a very high resolution, so we can know exactly what species are present in our soil. The problem is that it generates a huge amount of information, usually tens of thousands of different data points. So we're gonna find a great diversity and an amount of different types of microbes in the soil. The problem is that less than half actually are identified. More than half of the microbes that we're going to find are unknown species. So those are species that we haven't identified yet, right? So what do we do with that information? Is that really useful to go that far, right? Especially when we're talking about soil health, because soil health actually relates to the functioning of soil. So maybe it's more interesting to actually look at what the microbes are doing, right? And so there's other tests that we can run on the soil samples, like for example, looking at the microbial activity. And this is again a very rough estimate of how much the microbes are respiring. And what we do is measure the CO2 that is being released from soils. A little bit more complicated, a little bit more expensive, it's enzyme activities. And here we are measuring the activity of enzymes that are produced by microorganisms and that are being used in the breakdown of organic molecules. Like for example, protease, it's an enzyme that breaks down proteins. Cellulase, it's an enzyme that breaks down cellulose. So this is very informative to show how organic matter is being decomposed and how nutrients are being released. So enzyme activity is very well correlated with nutrient supply in the soil. And now a little bit more complicated, or actually way more complicated and also way more expensive, we have metagenomics, which is the analysis of functional genes in the microorganisms that are telling us the activity of catabolic and metabolic processes in the microorganisms. Again, this analysis generates tens of thousands of data points for a single soil sample. So again, it's a balance between how much information are we getting and how much is actually relevant, all right? So these are questions that we're working on right now and within the context of Vineyard soil health. And specifically, the graduate student Noelimar Gonzalez Maldonado, who's working in my lab, is working on this within the context of vineyard soils and in Napa Valley. And probably a lot of you know her already because she's been contacting you and talking to you over the last months. So her work is actually trying to define soil health for wine grape production, addressing all of these questions that I just mentioned in the previous slides. And for that, she's using a combination of semi-structured interviews with growers and vineyard managers, and a soil survey and soil sampling and analysis that we're doing over Napa Valley. So there is a social science component in this project together with the soil science. This is a project that we started last year with support from the American Vineyard Foundation and also in collaboration with the Napa Valley grape growers. And so we started right when the pandemic started. But luckily, because this project has two components, a social science and a soil science, we were able to kind of adjust things. So we started by doing the interviews that were mostly done online through Zoom. And so I'm gonna show you some preliminary results of these interviews. And actually, before the interviews, we sent out a survey to the growers to kind of assess, to gauge their interest in soil health. And so this is a little bit of a breakdown of the people that we interviewed. So there was kind of a balance between operation sizes. We had large producers and small producers in equal numbers. Also with different amount of experience. Most of them were vineyard managers, and about a little bit more than half were conventional. Some of them were organic producers. Now, something that stood out is that the large majority of the people that responded to this interview, to this survey, said that increasing soil health is a goal for them. All right, so it's one of their management goals. So after this, Noeli met personally through Zoom with all of the participants. We had up to, I think, 17 interviews so far. These were semi-structured interviews, and the purpose was, and the reason for doing these interviews was to be able to build a community-based definition of soil health. So we wanted to extract those variables that are the most important for wine grape production from growers, right? To get that information from them. So there were overall 20 questions, but those 20 questions were centered around these 6 topics that are listed here in this slide. So we asked about the importance of soil health for wine grape production. What are the properties of a healthy soil, or what is most important for producing grapes? What is the understanding of the definition of soil health for growers and vineyard managers? What are the practices that they are using, the benefits that they have observed so far, and what are the difficulties that they have experienced? And we also asked about what's the current understanding of the soil properties that are better related to soil health. And specifically, we asked about the understanding of the role of the soil biome, the soil microbiome on soil health. And so this is a preliminary analysis of that qualitative interview data. We recorded, transcribed, and then analyzed the information on those interviews. We're still working on the analysis, but we have run a preliminary analysis, which I'm showing you here in this slide. So what we've seen is that most growers consider soil health to be important for wine grape production. And also their interpretation of soil health is consistent and very specific. So growers associate soil health with lower erosion, higher vine health, bigger, and grape quality. All right, so there's the people that it's on the ground managing the soils and harvesting the grapes, they already have a good understanding of how important soil health is. Now, when we ask about what are the most important properties that we need to look for in a soil, then there were a few things that came up that were also kind of common across all of the interviews. One of them was balance, the concept of balance. And so when we asked about that, then it was mostly about physical and chemical properties of the soil, right? So in terms of physical properties, what growers are looking for is a specific soil texture that is balanced. So usually a loamy to coarse texture is preferred. In terms of chemical properties, growers are not interested in high levels of nutrients, but rather just enough nutrient supply. So a balanced supply of nutrients, right? It was also interesting to see that few growers mentioned soil organic matter and soil biological properties as being important in soil health. And we also identified that there's quite a polarized view on soil biological properties. So microbes are either friends or foes. They are the good guys or the bad guys, right? The last thing that we identified is that there's uncertainty in how the management practices that are currently being used, like tillage, cover crops, compost application, are actually influencing soil health and grape quality. And so this last point is what I'm going to go on in the next slides. What is the reason for this uncertainty? So in the presentation yesterday, we talked about how practices like compost application can have a clear and direct effect on soil health. We know there's research which are, there has been studies done over the last 20 years in California and other wine growing regions in the world that are showing beneficial effects of these practices for soil health. So this is nothing new. Now, the problem comes when we look at the outcomes of these practices in different places. And so for the same practice, what we're seeing is that the outcomes for soil health are absolutely different for different vineyards, different growing regions, right? So there's something there that we need to understand. How is it possible? What are the sources of variability that are causing these different responses? And so because we don't understand that, and it's really difficult to predict the outcomes of healthy soil practices for grape quality. So to solve this problem, we have to start with the first part, right? How do we solve or disentangle this variability in soil health outcomes? And I'm going to go over some of the reasons for that. First of all, an example on how the soil, different management practices have actually different impacts on soil microbial communities, right? So we know that cover crops are beneficial for soil health. They increase soil organic matter, they stimulate microbial activity. Now, depending on the type of cover crop that it's being used, whether it's a legume or cereal, mustard, different mixes, the microbial community that is observed in the soil can actually be extremely different. And this is a study that was published by Kyla Burns and Kerry Steenworth that was done in Napa Valley a few years ago. And so this chart is showing you these different data points are actually representing the diversity of the microbial community for a single soil sample. So the closer together, the more similar they are. The further away, the more different, right? And so the different colors are representing different cover crops or different cover crop mixes that were sampled in Napa Valley. So we got grasses, a mix of legumes and cereals, a mix of legumes, cereals, and mustard, legumes, mustards, and soils without cover crops. So as you can see, generally, those different types of cover crops, they can be teased apart. They are different from each other. The microbial community is different, right? So that kind of adds to that uncertainty, right? The type of management, the type of cover crop is driving the microbial community. So things get complicated, right? And the same happens because we've seen this in the literature with different types of compost. We know that compost and vermicompost affect the microbial community different, right? Whether this is important or not for wine quality, this is something that we need to understand. Another reason for these uncertainties is that typically practices are stacked on top of each other. They are combined, right? So we want to assess the effects of one practice on soil health or wine quality or grape quality, and it's really different to disentangle this, right? If we go to actual farms, usually cover crops, um, which can be different species of cover crops, can be combined with compost. Compost can be applied at different rates. They can be produced from different sources. We can have grape pomace compost. We can have dairy manure compost, right? And these two practices are at the same time combined with the tillage practice, right? Some growers till, some others don't till. And so while this is interesting, it adds a lot of complexity that makes very difficult to tease apart the effects of the practices. So this kind of reinforces the need for experimental approaches that are carried out under controlled conditions, right? So like the research that is going on at the Oakville station right now, it's extremely important. In addition to on-farm kind of real life research. And then a huge source of uncertainty is the soil type. Practices are applied to a wide diversity of soils with a very different soil properties like textures, right? And we know for sure that the response of those soils to management practices is also very different. Like for example, we know that texture is very, very important for carbon sequestration. So soils that have a higher content of clay will sequester more carbon and will store more organic matter than soils which have a coarser texture or a lower content of clay. And this is because clay particles are very reactive, right? They are charged, and so they tend to attach organic molecules, which are also charged. So it has to do with the chemical reactions in the soil. So this means that when adding compost, when increasing soil organic matter, the expectations for different for different soils, they should also be different. So this is something that has been observed again by Kerry Steenworth and Kyla Burns in Napa Valley. This was an assessment that they did 10 years ago, and they looked at just about one very simple indicator, which is soil carbon across different soils in the valley, and they've seen that it ranged from very high contents to very low contents, right? So how do we deal with this, right? We need to kind of establish benchmarks and guidelines for growers to understand how much they can increase their organic matter depending on the soil type that they have. And this is the second part of this project that it's right now being carried out by Noel Limar, the grad student, and our research team, together with Khan and Kerry Steenworth and other researchers. We are going back to the same sites in Napa Valley that were studied 10 years ago. We are collecting samples again, and this time we are doing a comprehensive analysis of soil health. And so the idea here is to create a range of variability for different soil types, for the different soil indicators, all right? So we want to generate information that will allow to know the lower or higher upper boundary of soil health, for example, soil organic matter accumulation for a specific soil, right? So in that way, growers will know what to expect when they are applying a specific soil practice, right? So if a grower is applying soil compost to a soil with a coarse texture, then that grower will know that they can only increase soil organic matter up to a certain point, right? And so hopefully this will help improve adoption practices, improve the adoption of these practices, right? Another thing that we are doing in this project, it's we are trying to disentangle what are the most important drivers of the soil microbiome. Given the key and central role of the soil microbiome in soil health, is the microbiome shifted or regulated by static soil properties like texture, or the amount of clay or the amount of sand in the soil? Or is it more regulated by dynamic soil properties that can be changed by management, like changing the carbon in the soil, right? So we are doing in the same samples that we are collecting in this soil survey across Napa Valley, we are doing a comprehensive analysis of the soil microbiome using the techniques that I explained before. We're doing analysis of PLFAs, and also recently we just got support from Biomakers, which is a biotech company focused on the analysis of microbial communities using DNA techniques. We're going to carry out analysis of fungal and bacterial communities using genetic markers. So hopefully putting all this information together will help us understand a little bit better how the microbiome can be managed. The second question that we want to address is how important is the microbiome for soil health, really? And so we want to run these correlations between the microbial parameters that we are going to measure and the indicators of soil health to hopefully find some variables that can explain changes in soil health. We also want to select variables, microbial variables that are informative, right? So it's PLFAs better than DNA markers. It's microbial biomass carbon enough to give us an idea of soil health, right? Also, how does soil microbial diversity relate to the functions on a healthy soil? Does it really matter, right? Do we actually need to look more for the functioning of the microbiome rather than the structure of the microbiome? So these are questions that we are working on. We are starting our sample collection next week, so really excited about that. And we will be working on analyzing the samples through this year, so we'll have results very soon. The second part of this question that I showed you before, so once we disentangle all that variability associated with the outcomes of management practices, then we can address the second part of the question. It's like, all right, how are these management practices associated to grape quality, right? And so that will allow us to define indicators, soil indicators of grape quality, all right? Now, one of the most important things when managing for the quality of grapes that was brought up by many growers in the interviews, it's the balance in nutrient supplies. This is something, a property that is desired by the industry. And so this is actually a property that is typical of healthy soils, right? A healthy soil should be able to balance the nutrients of the supply of nutrients and kind of provide nutrients to the plant as the plant needs them, right? So the objective of healthy soil management practices is to build a soil organic matter pool and a microbial community that releases plant nutrients as the plant needs them, but also retains and recycles nutrients, all right? So how does this look? In like conventional systems for like crops other than wine grapes, let's say corn. Let's use the corn example again. Fertilizers, I'm going to move this. I'm not sure if I'm, all right. Fertilizers are usually applied in big amounts. So the ammonium and nitrate can be supplied to the crop, right? And in the case of corn, that's a crop that requires high amounts of nitrogen. So then growing corn, it also requires high amount of external inputs of fertilizers. right? This is how things are managed typically, right? Now, things change if we want to manage for soil health. If we manage for soil health, what we do is building up the pool of soil organic matter so that supply of ammonium and nitrate to the crop will rely more on organic matter and less on external fertilizer inputs. That's the idea, right? At the same time, because we are providing a more gradual release of nitrogen, the pool of available nitrogen at a certain moment in the soil will be lower, and that decreases the losses, the risk of losses, right, through leaching or nitrous oxide emissions. Ideally, also, the microbial community in a healthy soil will use up all the leftover ammonium and nitrate so it's not lost. So the nitrogen is released and recycled within the system. Now, how do the things look for wine grapes as compared to corn? So wine grapes, as we mentioned before, they do not take as many nutrients as corn. So the input of fertilizers are lower, right? And the amount of ammonium and nitrate that are required at a specific time in soil is also lower. In a conventional system, this is supplied by fertilizers, right? And it doesn't rely so much on the organic matter pool. In a healthy soil system, the idea is that we build up the soil organic matter so that by mineralizing organic compounds, it supplies that ammonium and nitrate, but also consumes the excess of ammonium and nitrate, decreasing the losses, right? In this case, because the needs for fertilizers are substantially lower, then the inputs, externally inputs can also be reduced substantially. So how can we achieve this balance, right? There's two things that we can do. And for that, we need to take into account the source of organic matter and the type of organic input. And second, the soil microbial community, right? We need to balance the two things and understand how the two things are working. In terms of the properties of the organic matter, like let's say the compost that you're putting into the soil to build up the organic matter. One of the most important things that you can do to manipulate the release of nitrogen is understand and manipulate the carbon to nitrogen ratio of the organic matter. And I'm going to explain why. The thing is that microorganisms break down organic matter for their own growth, right? They consume it to get their own nutrients. So they will take up carbon and nitrogen in the same proportion as they have in their biomass. So let's say that we put an organic residue, compost, or organic fertilizer in the soil that has a carbon to nitrogen ratio of 50:1, meaning that there is 50 parts of carbon to 1 part of nitrogen. Microbial biomass, and specifically bacterial biomass, has a much lower C/N ratio. So it's usually around 8:1. So microbes will consume 8 parts of carbon for 1 part of nitrogen. Now, what happens when you put the two together? That bacteria, it's only going to consume 8 parts of carbon for 1 part of nitrogen. The rest of, so that means that there is a lot of residue which is going to be undecomposed. To be able to break down the rest of the residue, that bacteria needs a lot of nitrogen. And so it's going to scavenge and look for whatever nitrogen it can find in the soil. That leads to what we call nitrogen immobilization. That happens when the microbial community takes up all the nitrogen that it's available in the soil in order to break down organic matter. All right. So this is something that can be manipulated. And we know that there is a threshold of C/N ratio that actually results in mineralization or immobilization. And so very quickly, I'm going to show you, here's a summary of different organic matter, different types of organic matter, their C/N ratios, and whether they result in mineralization or immobilization of nitrogen. For example, organic matter or organic fertilizers with very low C/N ratios like blood meal, fish meal, and soybean meal, those are going to result for sure in a very rapid release of nitrogen and clear mineralization. If you go to the other extreme, you talk about straw, corn stalks, those have a very high carbon to nitrogen ratio. So initially they will result in an immobilization of nitrogen. Compost is kind of in the middle, right? And the CN ratio of compost can change. There's compost with lower and higher CN ratio, but typically compost will result in a gradual mineralization of organic matter. It's important to understand that this immobilization is temporary. So there's only so much that we are going to manipulate available nitrogen by using these. Typically, when we add a residue to the soil that has very high CN ratio, this is what we see with available nitrate, for example. So the content of soil nitrate in the soil is going to drop after we incorporate our organic residue. And so at the same time, there's a very sharp increase in microbial biomass because the microbes are actually incorporating that nitrate into their biomass, right? But as the organic matter is decomposed, then microbes kind of run out of a food source and they start dying. And as they die, they release the nitrogen back into the soil. So this is something that can happen within a matter of weeks. So it's temporary. How can we actually achieve a long-term balance between the organic matter and the microbial community and the nutrient supply to the vines? So this requires a more careful understanding of the microbiome and how it's affected by long-term inputs of organic matter. Okay, and so this is my last slide, and I'm going to show you projects that we have ongoing. So we have a trial going on. We actually just finished it at JLOR in Paso Robles, where we apply different rates of compost to a soil. And so what we're gonna do now is analyze the microbiome in those soils and how it was shifted by 3 years of compost application. And also we have bionutrition and grape quality data. So we're gonna be able to relate the properties in the soil to the grape quality. And then we're hoping to start next year a couple of trials. Those are trials that are actually already running. They have been running for 3 years. And in those trials, they're also incorporating compost and for at least 3 years again. And so we're going to hopefully, with the support of the CDFA specialty crop block grants, we are going to do a comprehensive analysis of the soil microbiome, the structure, the activity. We're going to measure soil health, grape quality in detail. And so, yeah, this is something that we hopefully are going to be working on in the future. So I hope I gave you a little bit of a picture of the things, the exciting projects that we're working on, the importance of really defining soil health for wine grapes and all of the things that we still need to understand. And if you're interested in working with us, especially now as we start the survey in Napa Valley, please send me an email. And so we can incorporate your vineyard into our sampling. And again, I would really like to thank to the American Vineyard Foundation and the Napa Valley Grape Grower Association for their support.
Okay, great talk as usual, Christina. It's very informative. And then I guess it's safe to inform the panel that the project is expanding to the Central Coast this year, correct?
That's right. Yes, yes. We just applied for more funding to expand the same idea to a different region.
Not enough, in my opinion. Not enough. So, anywho, but yeah, so if you are interested in participating in these, please contact Christina or get a hold of me. I can get you in contact with the correct person to participate in the survey through Christina. So anyways, we have a few minutes for questions that I will try to take, and then we're off to a deserved break, I think. So the first question, I will start from the bottom.
Okay.
Um, First question is from one of our former farm advisors, Maxwell Norton. And this is my question as well, and I can supplement the answer to this question. Most of the grape acreage is in the San Joaquin Valley. That's where wine grapes are grown within a 100-mile radius of Fresno. Why don't you do the survey there?
This is a great question, and I actually thank you for that, because I didn't explain in my presentation that what Ken just mentioned, that the idea is to expand this same project to different wine regions, wine growing regions in California. So next year we want to do Paso Robles, and then on the following year we want to move to Lodi, and then hopefully we keep going. And because there's only so much we can do in a year, but then it's extremely relevant to study how these questions are actually different in different areas, which have different growing priorities, different soils, different climate, etc. So there's a lot to cover. Yeah, and we would love to work there.
So to supplement that question, we have a privately funded study where we are looking at the soil microbiome in west side of Fresno in salt-affected areas. That's irrigated with winery wastewater. So we have a graduate student that's looking at that. But analyzing microbiome data takes about a year and a half. It's a slow process. Nothing is quick. So anyways, so I have a question from Patrick, who's asking, isn't the pH the only soil attribute that's consistently related to microbial diversity?
That's a great question. Globally, yes. So there has been recent studies that show that of all the things that can shift microbial communities at the global level, then pH is one of the most explanatory soil variables, but it's not the only one, right? And when we manage soils, we actually change the pH quite a lot, right? And not just that, but also carbon, etc. So we still need to understand a lot about these things.
Uh, Anonymous attendee asks from Modesto, what is the timeline for nitrogen immobilization after organic fertilizer application?
So it can happen usually within weeks to maybe a month, but it's short term. It depends on how much organic matter is decomposed in the soil, because the microbes kind of eventually die down right after they consume everything. They die and they release the nutrients. So it's temporary.
Yes. Question from Amador and El Dorado counties. Why doesn't anyone do studies in Amador and El Dorado County?
Yes. We just started again. We would love to move to other places, and we hopefully are going to do that in the next years. Yeah. So what we learn, we're learning lessons from the study that we're doing. And so hopefully we're kind of improving and fine tuning our research questions so we can actually move to those areas. But the idea is to do that.
Yeah. There are lots of questions, as usual.
I can try to address them also,
afterward. So here's one that you're gonna get over and over. And this was also in a Netflix documentary. So can you relate the results of minerality in wine with compounds in the soil?
I didn't understand the question.
Yeah, no one does. So there's this concept of minerality in wine, and then is this related to the compounds in soil?
Well, that's what we want to find out. And I don't know exactly what minerality is. And I have to say that I'm a soil scientist. I'm just starting to learn about wine grapes. But our research is trying to find that connection between the soil and, what you can do to the soil and the grapes.
Yeah.
in terms of nutrition, right? There's also water is very important, as Ken knows, and so that will also affect the grapes.
Yeah, I mean, Nazareth's going to talk about that later on in the day, because we are getting into these hyper arid cycles that's affecting grape composition greatly,
Mhm.
and not in the manner that we would like. Even in Napa County, where we're accustomed to seeing 1,200 millimeters of rain, we're seeing about 250 millimeters of rain. I mean, it rained less than Fresno County and Napa County.
Mhm.
So, anywho, so here's another question. University of California, Davis professors taught me that organic matter content of the soil was historic and cultural practices were not going to significantly alter it in the long term. Has this perspective now changed?
Well, there is a part of that that it's true, so they didn't lie to you. But we know that practices can change carbon really quick in the short term, and we have seen that, for example, in the delta soils around here. You know, subsidence. That's something that just happened in a very short period of time. Those are soils that take forever to build up. So, yeah, long-term organic matter accumulation can take a long time, but it can also change quickly depending on the management. And you know what we've seen is that our trials, depending on what fraction of carbon you look at, you might see a stronger effect or not. So if you look at the overall carbon pool, you analyze total carbon pools. That's it's something that it changes very slowly with management. But there are fractions of carbon, like labile carbon and active carbon, that change really quick, and they have a very strong effect on the microbiome, which is then, you know, releasing nutrients, etc.
Yeah. So to follow up that, in your talk, you mentioned soil organic carbon and soil active carbon as soil health indicators. Please explain the difference.
Oh yeah, okay. So total carbon includes all the different types of carbon in the soil, and that, you know, it's like from cellulose and lignin and things that are very complicated. They have very complicated molecular structures, um, difficult to break down, to all the way to things like sugars, amino acids, and things like that that are very easy to break down. So not all the carbon in the soil is the same. And when we talk about active carbon, we usually talk about the things that are easy to break down, that are going to be used by microbes. And so it's more relevant for agronomic purposes.
Yeah. All right. I believe we have gone over our time. We need to be on a break. I
right.
will see you guys back in a few. Our program is going to start again at 10:30 a.m. So we'd like to thank our extension partners again. If you'd like to participate in our extension partnership program, please contact any one of us in the department.
Is Christina going to answer some of the questions in the Q&A?
That's the hope.
Are you still there, Christina?
Yes, yes.
If you can, if some of them, you know, if it's possible.
I'll do my best.
Oh, great. Thank you very much.
All right, let's go on break. See you back at 10:30. Okay. All right. It was a short break, but we are back. And my cat is like carting on the dining room table. Anyways, our next speaker is Professor Andy Walker from our department, from viticulture and enology, who probably needs no introduction. But so without further ado, I would like to turn it over to him if he is ready to talk about the.
I'm ready, I think.
Okay.
Can you see me and hear me?
Yes.
And you know, my presentation's up and rolling?
No, it is not up and rolling. So
No, it's not.
You'll have to share your screen.
Hold on a second.
There we go.
Can you see it now?
No.
No, we're seeing you as a spotlight.
I can't get down to the share screen for all of a sudden. It's not working here. Where's the image? Hang on a second. When in doubt, click out, right?
There we go. You're muted, Andy. Here we go.
All right, how's that look?
Perfect.
Good.
Okay, I've got you guys right in the middle of my screen. Let me move them out of the way. All right, so Con asked me to talk about nematode-resistant rootstocks, and I guess the role they're playing in healthy vineyards as well. And so I'll try to cover that sort of stuff as we go along. My research has been funded for quite some time by the California Grape Rootstock Improvement Commission and the Research Foundation, and the Fruit Tree, Nut Tree, Grapevine Improvement Advisory Board, and the Tableware Commission, and the American Vineyard Foundation, and Ian J. Gallo Winery, and the Rossi Endowed Chair of Funds in Viticulture have really been a tremendous benefit to getting all this work done. And it's going to start again, right? I'm retiring in June, and we're bringing on a new breeder, I hope, and that'll be a smooth transition. So a lot of the stuff today could be actually moved into the next realm of things as well. And I'll still be around to pester people, but not to work as much. So nematodes, are there soil-based interactions with nematodes? And that's a popular belief in the literature that certain soils are more prone to nematode problems than others. And it's true, but there are soils that are really agricultural soils rather than some sort of texture soils. But most of the agricultural soils are coarser textured from that perspective, sandier, preferentially over siltier, I guess, as you go along, and how they're packed and how they're broken up. In viticulture, we use other things, but we don't really see associations of soils and nematodes. So you can see, for instance, root knot nematode is on sandy soils only. Well, no, it's all over a lot of clay-based soils in the North Coast. And how did it get there? Well, it came up on planting materials primarily. So we move these things around the state on plant materials, and we need to be very careful. And that's part of the restrictions of the program of the certification program is to make sure these materials are free or treated for nematodes as well. So there's an effort made, but they're very small organisms, and it's very easy to move them around and get contaminated as this goes along. So sites that are previously in agriculture are much more likely to host grape damaging nematodes. When someone asks me questions about what rootstock do I use, the first questions I ask them are what was planted there first, and how long ago, and what's the potential for having cross-contamination with whatever the original pest was. And it's usually pretty high. And native vegetation sites, it's rarely a problem. Either the grape damaging nematodes are at such low levels that they're not problematic in the first generation, or they don't exist there to start with. So it's an interesting problem to look about and think about how those things have evolved and where they've moved over the years. So one of the primary tenets of agriculture is fallow. Fallow and crop rotation are the ways we control soil-based pests like nematodes, and they're very awkward with grapes. First of all, grape roots don't die very quickly. They last a very long time. tremendously long time in the soil, and no one knows how long. But I would think upwards of 10 years is not a poor guess. So that makes fallow difficult, particularly if it's high value land, which it often is in viticulture. And we need to leave it out of plantings for some period of time, out of grapevines for some period of time. So it's an issue, and we need to know what to rotate into, and hopefully it's a high value crop. We have almonds now, but they also increase nematode pressure in soils. Grapes following almonds is probably not like a great idea in terms of reducing the pest populations in those situations to start with. And that fallow again has to be long-term, and we can't afford the long-term basis of it oftentimes. So not only do roots last a long time, they persist and live. They actually grow in the soils without top growth, and they're able to survive and produce callus and new rootlets, and those support nematodes pretty aggressively. And nematodes also insist. They go into these hibernation stages and last for very long periods of time. We had an ice chest or a cooler full of soil and Ziphium index, the dagger nematode, and I had a student working on it and was able to extract nematodes out of that soil, completely bone dry, no growth of the plant material, no roots in it for that matter. It was really pretty clean. And those nematodes were still alive after a year and a half. And then the French said, we can do better than that. And after 4 years, they've done the same sort of thing, discovered that they'll survive. So it's very hard to avoid them once they're there. So what's the solution? The solution is resistant plant material and rotating that material as well. So if we're using a rootstock that resists nematodes in generation 1, we don't want to use that same rootstock in generation 2. And we'd be best off in breaking the pest adaptive cycle and leaving that out for some longer period of time as well. So, or switching them with even a more diverse series of rootstocks as well. So that's a key aspect of avoiding these problems. This bag here shows that the 6 years on a rocky terrace up above Lake Hennessy in Napa. These are finger size and smaller roots, and we dug down through a site that had been cleared and eliminated of grapevines for many years, just a rocky terraced area. And we dug through it and brought these roots back, and I stuck them on my desk. And within 3 months, there was growth in those bags. And within a few weeks, there was moisture in them. So something was respiring, probably more than just the soil fungi. And in fact, it was the roots that are alive. And you can see the little rootlet in that picture there too. So it's not easy to avoid these things. And how much root pulling is a good idea, and how expensive is it? And can you get rid of all the root pieces? And the answer is often no. And yes, it's very expensive. And no, you won't get rid of all those nematodes necessarily. So what are the effects on grapevines? Well, it's the feeding damage, and that damage kills root tips and encourages more root activity oftentimes that proliferates around them and encourages attack of those roots as well. It disrupts the vascular tissue, so it slows down the uptake of water and nutrients, of course. And it's a whole lot like phylloxera damage. It looks like phylloxera damage oftentimes, and the effect on the vine is a lot like phylloxera damage too. So those of you who have lived through the phylloxera crises in the 80s and 90s remember it well. And one of the first things that the winery saw was potassium deficiency in all of those wines coming off of those roots. And again, it was because off of those affected grapevines on AXR. And it was, again, the lack of root activity and fine hairs taking up nutrients as well. Those roots crack and they gall. They gall first and then crack and split apart, and then they end up with you end up with decay and loss very rapidly. And there's a few of the vector viruses too. The dagger nematode group and a few others are pretty effective vectors of grapevine viruses. Most notably the grapevine family virus, and we'll talk a bit about that in a while here as well. So how do you sample for nematodes? Frequently, I'll get samples in, and they're from areas we know that have severe pest pressure and damage, and we can't find any nematodes. And so it's a pretty difficult process to begin the analysis of what's there if it's not there in the first place. So where do you sample? That's the key. And you often are told, well, random sampling is an effective way of going about this, and it's not at all. If there are no roots, there are no nematodes. So you need to find those nematodes first. You find those roots first before you can find the nematodes. And it's also not an easy task, particularly if you're dry farming. If you're on a drip irrigation, it's much easier, and you just go to that drip zone and you can actually follow those populations pretty effectively in those situations. So then the question becomes, do you take the samples from healthy vines or diseased vines? Diseased are easier, right? Because there's less growth, fewer roots to impede the shovel. But there are also very few nematodes on the diseased vine. They've already left those vines, the damaged vines and weakened vines. They've left those vines and they've moved to healthier vines on the periphery of them. So it's always best to find the holes, these spots where the feeding damage is more intense, and then go to the periphery and find vines that are fairly decent in terms of their growth and take your samples from there and in the drip zone. So we remove the surface soil, take off about 6 inches of the surface soil, take the next 6 to 12 inches and put it in a plastic bag, and you have a sample. Okay, and other words to the wise are, don't let that sample bag get hot. It doesn't take long to cook something in those bags. And the nematodes are mostly extracted, most of the parasitic ones certainly are extracted through their ability to sieve out nematodes and take the ones that are alive and sample those. And the ones that are dead don't get sampled because they don't wiggle through the system very effectively. So it's something that's a key to really get those samples off to the lab quickly and keep them cool and really respond to that. So if you're sampling, find the diseased vines, go to the healthy vines and near the diseased vines, take those surface roots more or less in the drip zone, and you'll get a good indication of what's going on there. The questions you'll be asked are, what was the previous crop? So it helps to know that. Annuals versus perennials is critical. There are more perennial-like nematodes that are more aggressive on grape and trees as well too. So it's good to know what that crop history was and certainly what was there ahead of time. If you're going to fumigate, and there's an effort to bring back other more gentle forms of fumigation, but one of the key issues is, how do you get the soil dry enough to get good penetration? So when you actually apply these fumigants, it needs to be dry if you're going to use them. Those soils need to be dry, and it's really difficult to get them down very deep. So their efficacy is not great. They work beautifully for annual plants because you don't really have to worry about a very deep root system, but with grapevines, that root system gets down and out. It's difficult to get good efficacious coverage. So what sort of nematodes are we pursuing? I'm sure Andreas talked to you about this, so I'll just sort of gloss over a little bit of this as we go along here. But there's ectoparasitic types that feed from the outside of the root, that feed with deep styloids that probe into the vascular tissue of that root. And there's endoparasitic nematodes that actually migrate inside the root, bore themselves inside the root, and migrate through those roots and damage in that sense too. So there's two different sorts. They cause similar sorts of damages, but you can tell the difference if you think about how they're formed. So if you look at that root tip and the root mass is sort of in a clustered wad of roots almost that really don't move out of there, almost like a Brillo pad, as opposed to having knots or beads or galled lesion tissues on it. Those things will help if you look at that. Look at those roots when you're sampling to get some sort of idea of what you're looking at too. So with fan leaf, we have it all over the country, all over the world. It's the most important grape nematode, probably from that perspective. It doesn't cause the most damage, but it does vector a virus that's even more insidious than the nematode feeding oftentimes. And that's grapevine fan leaf virus that prevents proper set of the clusters and great yield declines. That, interestingly enough, it's much less severe. Both the nematode incidence, the nematode damage, and the virus incidence and the virus damage is less severe in hot climates, which is quite interesting. You can get pretty good indication that the virus is present, but usually the response in terms of yields and fruit set is much less damaging. So we tend to overlook that in those cases and not think of it as being a very serious problem sometimes. But it's certainly much more severe in cooler climates. Okay, we've talked about this already. We do have one rootstock alternative. It's 03916, which is half vinifera and half rotundifolia. Remember, I hope you remember that we don't use vinifera in rootstocks because it invariably is susceptible. But we've now had over 30 years of commercial use of 03916 in vineyard situations around the state, and it has not collapsed, which is great. Its sibling, 04343, was released at the same time, and it collapsed the phlox in 2 years. So we're always a little bit nervous about recommending this rootstock and not leaving any caveats about its use. But it's survived to this point, and it looks interesting. Now, the next question is, what do you replant it with? And that's been an interesting process too. This root system is definitely very resistant to the feeding of Xiphium index. It doesn't feed on it in a lab-based sense. We don't get reproduction on the system, and we don't see damage on the root system. So what happens to the roots on these vines if there's no Xiphium feeding on them? they flourish, of course, they do fine, and the nematode is dying off. So it might be possible to look at O3916 as a natural nematicide or almost a means of controlling this problem in one generation. We have a lot of questions about its effectiveness and whether it's a good idea to be using it in the first place, because it tends to be excessively vigorous oftentimes on deeper soils. But it resists the disease, and it may be that we could use it a generation and pull it and replant again. We did some testing on that. We did some in some old rootstock plots that we knew we had at least 15 to 24 feet between the vines, between the rows of vines that were infected. We dug down deep with backhoes and we searched for nematodes, and we found a few, not many, certainly hundreds and thousands in the susceptible plants, but very, very few, if any, in the O3916. And the question always then becomes, are they feeding on vinifera roots, this generation of roots, the last generation of roots, or are they feeding on roots from lateral penetration from other rootstocks in the trials and areas? And we haven't been able to determine that at this point. And it'd be a good, nice, important study, I think, over time, but a lot of work to go through. There's Ziphium americana, which is also called the dagger nematode. So it's good to distinguish between these two, because this one doesn't really cause a serious problem in California, we don't think, but we're not sure. So here's a nematode that's definitely capable of causing damage to the roots of grapevines. And we don't really know much about it because we can't really cultivate it. We haven't been able to grow it in pots in a greenhouse environment or another test system environment. It's very hard to study these things in the soil oftentimes, and it's hard to pick them apart in terms of which species you're looking at sometimes. And this is a very complicated species. Species is probably a complex of as many as 8 or more different species that were all bundled into the same pot. But in general, we don't worry about it, and maybe we should. And that's for my successor to figure out if we should worry about those sort of things over time. But it's been very, very difficult to really study it fully and understand its potential. Root knot nematodes have been generally associated with further down the valley sites, sandier soils, more agricultural soils, not as viticultural, where you have rapid turnover of your rotational cycle of what you're growing. And that helps discourage the pest, but it also is helping them diversify too. So in a sense, it becomes a long-term system where it really becomes necessary to have some means of knocking back these nematodes over time. And I'm sure Andreas told you lots about that, so we won't go much into it as well. But they can be very, very damaging and very, very effective at controlling growth of grapevines and really stunning back the root systems entirely. One of the most important nematodes is the ring nematode, particularly in a replant sense, because it builds up on woody plants if you're following with woody systems. It doesn't cause much galling, but it causes a lot of death of that root tip, the fine fibrous roots, and it stubs those back, and the plant produces more roots beyond that point, and then it stubs those back as well. We think of it as being mostly a replant problem, because we'll sample that the existing vineyard where these vines have been declining in the proper way, and we can find thousands of nematodes and many thousands in some situations, and the original vineyard has not been that badly stunted, and the next one goes in, and of course it fails in those sort of populations. So it's a nematode that can be outgrown, I guess, and an older vine is able to outgrow or sort of defend itself by pure growth. But a new plant in a potted vine situation is really under a lot of pressure and don't do quite as well. We have two sources of resistance. That's the good news. The bad news is they're both rotundifolia-based rootstocks, and they're both difficult to propagate. And we just mentioned O3916. We'll talk about GRN1 in a bit here. They both are very resistant to ring nematode, which is great. But they're both difficult to propagate, and the quality of those plants can be uneven oftentimes from nursery sites too. So it's a bit tricky. There are ways to get around that. There are tricks to apply to getting these things to propagate properly, and it's possible to make them work, but it's more difficult, and it takes more time and more patience oftentimes. The damage is even more severe when we combine this nematode with other nematodes as well, and that often happens. We have these nematode complexes that form, and it's pretty ubiquitous on many different soils, and it's something you should really be aware of before you replant casually. And the question is, how do you get rid of it if you're not going to use O3916 and the GRN1? And the answer is, we don't know yet. We don't have a good feeling for the resistance across other species and across other rootstocks. Certainly the commercial rootstocks are not resistant to it. So it's something that we need to keep in mind over time. The legion nematode is one that follows woody plants again too. So orchard crops, particularly apple and pear were a problem as well. There are some problems down in other nut trees down the valley. And it's a nematode that attacks grapes pretty aggressively too. The selection that we use here on campus to screen, that I use to screen for legion nematode resistance is from walnut, and it does beautifully in walnut too. So it's pretty easy to keep track of it that way. Primarily found in sandier soils, again, more associated with a broader range of tree crops, and it can be a difficult nematode to avoid and have good resistance to. The citrus nematode is from the San Joaquin Valley, lower San Joaquin primarily, but it's moved up on nursery stock. We see it around occasionally. It's fairly damaging. It really has a fairly aggressive feeding activity, but it doesn't really seem to spread as quickly as some. And it's really been mostly a nursery problem that's migrated around the state in most situations. So what are we trying to do with breeding? What have we been trying to do from breeding all these years is make better resistance to nematodes, more broad-based nematode resistance. And now we've been building off of that to a lot of other things over the last 10 years. And one of those things has been salt and drought resistance, of course, at the same time. And whether or not we can effectively breed for those things and select for those things and combine those resistances or tolerances, if you like, to the resistance and under nematodes. So that's been a big effort is to utilize these GRN rootstocks we're going to mention in a minute here to have a broader range of characteristics, not just nematodes, but also maybe drought and salt if we could. Remember, in droughty environments, particularly in dry summer climates of the world, salt problems can be very severe. And we use water to leach and flush the soils. It has to be high quality water, and we need a lot of it. And those things are going away. We have less and less availability for that sort of water. So we're going to need better salt resistance, salt tolerance in our rootstocks. We can get our ring nematode resistance from rotundifolia, but can we propagate them? So we've been working a lot on trying to optimize that. I've been working on virus tolerance, particularly family virus tolerance, for a very, very long time, and we have a few little breaks in the activity here where we're finding some better forms of resistance. It looks like maybe tolerance to fan leaf. So we're moving forwards with that. Of course, we've been doing genetic mapping to look for markers to allow us to stack and combine these traits more effectively. So we don't have to test them as often. And in general, for marker selection, this MAS stuff, we don't really think of it as a selective tool, positive selection tool. It's a negative one. So it finds out what plants will not have that resistance or will not have that trait. We can discard those very, very early in the program and emphasize our efforts on promote our efforts on the stuff that has the markers, has greater potential of having the traits that way. And it allows us to stack these things together more effectively and more easily too, particularly when these characteristics are similar. We've been putting in a lot of campus rootstock trials over the last 5 years, 6 years, to look at these things sort of in sight here, where we can really look at their viticultural attributes a bit more, and maybe do a better job of getting rid of some of the stuff that really doesn't grow all that well. So rootstock development began, of course, with phylloxera 150 years ago almost, in that vicinity, or maybe a little longer in some cases. And we used that, the resistance to phylloxera that was found in the North American species to form the foundation for rootstocks in Europe. So phylloxera wiped out grape production over 30 plus years, and we ended up reestablishing with the resistant rootstocks that were all from American species. And of course, phylloxera is an American pest, so we have resistance to it. The problem is we don't have any rootability in the American species. They root very poorly from dormant cuttings, except for Riparia and Rupestris. So people often ask, why do we have so much Riparia and Rupestris in the background of these rootstocks? Why don't we use something different? And the answer is because we have to. We can't have the commercial levels of rootability without having these two species in the background somewhere. But they lack Lyme tolerance, and that's what introduced Berlandieri into them. So we couldn't have pure Berlandieri rootstocks, but we could have Riparia-based Berlandieri or Rupestris-based Berlandieri materials. That was key in this whole process, as we developed rootstocks many years ago. And so what have we done since then? Nothing. That's sort of interesting. That's not completely true. There have been a few rootstocks that have been put out over the years, but in general, all the commercial stocks we use have been bred very, very long time ago. And it's there's a few exceptions, but even those have some more easily rooted species that are related to the riparian rupestris in the backgrounds too. So what else were they selecting for initially? Of course, most of this work was done in conjunction with nurseries or by nurseries. So they were looking for long canes. They were looking for decent diameter growth. They were looking for limited lateral production. They were looking for early ripening of those canes and scions. And all those things were really selected for. Maybe not as intentionally as unintentionally, but they were definitely selected for and came through that way. The adaptation to wet or dry conditions was also obvious. There were northern nurseries and southern nurseries, and again, the advertency, whether they were looking intentionally or not, was sort of the issue here. But they were breeding in those environments, so they selected those things. And we have a series of stocks we consider to be more moist, northern European versus stocks that are drier and more Mediterranean-based, I guess, from a rupestris-based side of things and a drier-based side of things. They looked for larger diameter roots and shallower fibrous root systems. Again, not as intentionally as we would have thought, but that wasn't the most important feature. What they were looking for really was vigor and strength of those plants, and that was completely associated with the diameter of those roots and the fibrosity of those roots too. So the rooting architecture we're still looking at today, and we'd love to be able to define it more clearly, and we'd love to be able to select for it and classify rootstocks in different categories that were obviously going to be higher and lower and medium vigor, but it's been hard because there's so much variation in this trait. So it's difficult to really pin it down sometimes. But yet, I can select for things. I've been making crosses recently to answer the desire of some for extreme vigor rootstocks with very deep penetrating root systems, and it's possible. Some of these look more like carrots than grapevines, but it's possible. This slide appears not to memorize or look at, but it's interesting. So we did a big study the last couple of years, and we looked at all the commercial rootstocks here and in France. And we shared samples with the Montpellier collection, and we got some idea about the parentage of these rootstocks that we're using today. And we knew we had a limited genetic base in terms of species: Riparia, Rupestris, and Berlandieri almost entirely. But what we didn't know was that the Riparia selection and the Riparia selection and the Rupestris selection and the Berlandieri selection are almost all the same. There's a very high percentage of the exact same genotype that went in with these materials. So the Rosigier number 2 here is a Berlandieri type. It's in a large percentage of those. There's not a lot of other sorts of Berlandieri used. The Rupestris base is really mostly Dulot. It's mostly St. George. And we don't know the base of some of these things either. So they don't have any extinct or extant individuals representing the species that were used initially too. And then the Riparia Gloire was almost all the Riparia that was used. So why should that concern us? Because we need to have a breadth of resistances from a broad-based background to prevent the evolution of more aggressive strains of pests and diseases in the soil. And we'd be able to track and utilize these things more effectively too for different sites. But it was sort of as Jim Wolpert would have said, a come to Jesus moment where we suddenly realized, wow, there's not a lot of diversity here. Maybe we should be more concerned about how these things are going to survive over multiple generations from now. Maybe because we need to start, of course, breeding for future situations 20, 30, 40 years down the road. And maybe we should be breeding more intentionally for phlox resistance, for instance, where we'd have to, where we can maybe avoid some of these genetic bottleneckings that are occurring and the impact of them. So the groupings, we've talked about rootstocks as groupings and parentage groupings, and the Riparia Rupestris group was the initial one we had. And it was really to help strengthen the Riparia-based stocks. Rupestris was stronger, and it grows in rocky, gravelly creek beds, where highly erosive beds, where it needs a very deep penetrating root system, and it has that. So they were selecting for both the Riparia characters, which were usually earlier maturity, and the Rupestris characters, which were strength and vigor, and trying to find those two things together in the same background. And the most popular ones we have still in use today are the 3309C, which has pretty poor nematode resistance. In fact, it's one of our primary susceptible controls, and 10114, which is fairly good. But they're different in other ways too. It turns out that 3309C has fairly deep roots compared to a lot of the members of this cross. And 10114 has very, very shallow roots. So if you look at these vines, the 3309C looks like a Rupestris. It's a hybrid, clearly, and And we've proven that genetically, of course, over time. And the 10114 looks much more riparian-like, but it's also a hybrid. So there are the two extremes of what you would expect if you crossed riparia by rupestris. A bunch that's more riparian-like and a bunch that's more rupestris-like. So you can use that if you like. In fact, I don't tend to recommend 10114 because it really is very, very shallow, and it doesn't have any buffering capacity for dry years. It really is very dependent upon applied water. They both have pretty poor lime tolerance, and you can see that if you use them down in the Paso area or further south in Santa Maria, where a lot of limestone soils are prevalent. If those stocks are used, you'll find out where those lime deposits are in those soils, just by looking at the growth of the plants as they go along. And they're relatively tolerant of wet soils, which can be useful as well too. The Berlandia rupestris stocks were mostly Southern European and mostly more drought tolerant, and the intent was to get more drought tolerance. They in general have pretty poor nematode resistance. So they don't really have a broad base for nematode purposes, but good phlox resistance. They have deep root systems. It's coming from both rupestris and Berlandieri, and the lime tolerance is coming from Berlandieri. The 140 Ruggieri is sort of unique. We haven't used it much over the last years, but it turns out to have excellent salt tolerance, extremely good salt tolerance. In fact, by far the best commercial levels of salt tolerance stuff we looked at, and we'll talk a bit about that later on too. On the downside, it's harder to propagate. It doesn't grow abundantly. It's very shrubby and more like rupestris in its growth pattern. So it's not as commonly found or available in nurseries either. The Berlandia biroparia group was and still is the most widely used group of these materials, where you had the Berlandieri lime tolerance and you had the riparia cane growth, which was exceptional. You have the lack of laterals, which is exceptional. You have the earlier maturity, which is a positive thing, a necessary thing to get plants out of the vineyard and into the nursery. So there's all sorts of attributes you have to look at the same time when you're going through these things. Relatively good lime tolerance, and we haven't used them as much. There's 428 that's been used quite a bit still, particularly in the sort of the high end of the market, more low vigor and high intensity sites, I guess you could call them. Things like 5BB have sort of dropped out, even though they root well, they graft well. They have good general nematode resistance in 5C and SO4 too. Not much else in all the other representatives that we've looked at. Borner rootstock has sort of come and gone, so it's very difficult to even find that stock, but it had excellent nematode resistance. It was just virtually impossible to propagate. 1616C is a little different. It has Celonis in the background, and Celonis is a hairy form of riparia, I guess you could look at it that way. It has a little bit of candy cans in the background probably, and some riparia from the Northeast, and it exists in northern Texas up through the Midwest. And it has excellent nematode resistance occasionally, not always, but occasionally. It has very good salt tolerance, not always, but occasionally. And it's moderate vigor. And we find in California that its phlox resistance is great, and its nematode resistance is pretty good. It's middle of the road at least. So it's something in that direction. And it's fairly broad in terms of its resistance as well. It tends to be tolerant of wet, waterlogged soils we've seen in the past, on clay-based soil, heavy clay soils. And it's definitely a lower vigor plant if you're looking for that as well. But good, again, general nematode resistance. Most of our nematode resistance is coming from Vitis champignii in our commercial materials. It's a species that grows through central Texas and across to the east side and back a little bit to the west side of Texas as well. It has very deep roots. It's an excessively vigorous grapevine. It's a tremendous grapevine. It pulls down trees. So most grapevines sort of are shaded out by trees over time and they die off. But candy can solves that problem by getting so large that it'll just pull the trees over. It tends to ramble and grow for miles, perhaps in some cases. So it's an amazing plant. And it has pretty good nematode resistance and relatively good phlox resistance. Not as great as some of the other species, but pretty good. But the problem is its rootability, and it's not easy to root because of that champignii background has Vitis candy cans in it, and candy cans is hard to propagate. We don't see this plant anymore. It's very difficult to find in the wild. It's an interesting thing that seems to be going extinct as the hill country of Texas becomes populated by people instead of grapevines. Vitis sedicerfolia is really the part of that 1616C that we're concerned with, and a fair amount of variation here too. It grows in gravelly gullies and ravines. It clearly must have some fairly deep roots and good strong ability to survive dry summer areas and fairly wet winter areas in some cases too. But it has variable nematode resistance. We've used it in breeding a fair amount, mostly for its salt tolerance, not for its nematode resistance. In Dogridge and Ramsey is originally was incorrectly originally called Salt Creek. It is Ramsey. Salt Creek is a form of Duaniana, another species, but they have similar characteristics. They're both Champigny based. They're both very vigorous and excessively vigorous occasionally. And we moved away from them because of the excessive vigor and the difficulty in managing canopies. Even in situations where we want a larger canopy, they've been hard to really slow down. So that high vigor is it's a known, so you have to use it appropriately to avoid problems with fruit quality and over vigor and fine balance as well. Harmony and freedom sort of derive from that. One half of their parentage is 1/4 is Dogridge, and it's an open pollinated Dogridge, and we have also some Champigny in the background here from that. So these root pretty well. We're selected for good rootability. They don't have great phlox resistance. And in fact, Harmony fails pretty abundantly on the right soils. Freedom goes down on heavier soils too. We generally don't use them on heavier clay-based soils that are associated with phlox or damage, but it's good to know that and to use them appropriately. They're both pretty good for nematode resistance. They're sort of in the moderate range against most species. So there still has some utility, but we've seen the evolution and development of more aggressive strains, particularly root knot nematode, on Harmony and Freedom both. In fact, we use them in our breeding program to make sure we're getting severe enough challenge along these backgrounds too. So again, if we're going to continue to use these stocks, they probably need to be rotated with something else. So we break that selective pressure that's driving nematode aggression in vineyards. Well, 3916 was bred by Olmo and was selected and sort of finished by Leiter and Goheen here many years ago. A vinifera by rotundifolia complex, and always a little bit disturbing because of that vinifera background. Since it's half vinifera, we'd expect it to collapse. And again, its sibling 043 did. But it's again our only source of resistance to Ziphinema so far that's commercially available at this point. We have some new ones that we'll talk about here briefly, but they're less available or less widely used at this point. Not very good root knot nematode resistance, and it can be affected by root knot nematode in places like Lodi and the Upper San Joaquin, where we have both Ziphinema and both Ziphinema and root knot Boledogyne species in the same soils. It's a bit high vigor in terms of its sign growth, but it responds well to deficit irrigation. You can cut it back pretty quickly and slow it down pretty aggressively if you're careful with the irrigation. And you can put on a larger trawl system, and you can manage that vigor as well. So it's one of these necessity stocks. So on a fan leaf site, it's very difficult to avoid having to use this at this point. So we've been looking at Muscadini rotundifolia for a long time. This rotundifolia resistance is amazing to everything. Unfortunately, it doesn't work very well in terms of propagation. It doesn't propagate most of the time, which is an issue. And it tends to be very tropical in its growth habit. It's a long cycle growth. It grows right up to the frost and beyond sometimes, and it can be problematic. And its bloom system is not well synchronized with other species, so it's hard to get it crosses it. It's a peculiar problem, but it resists virtually everything that bothers us. That's the interesting part about it, the interesting part of the quest, I guess, as you look at it. I put this chart in here. You can look through it later on if you like, but it gives you an idea of the vigor of these things. So how do you choose a root knot nematode stock or any nematode stock? Once you have some idea of its nematode resistance, do you need a high vigor one or a low vigor one? And this gives you some idea in this chart here. In the upper left-hand corner on the top row is the most vigorous of these materials, and the lower line in the right-hand side, the Ripariglor, is the least vigorous of these things. So it gives you some idea of how to utilize them and how you can mix and match in some spots. And so again, we've talked about why do we worry about nematodes? Oftentimes we don't know about them until it's too late. And it's almost always in those replant situations where you haven't really done a good job of analyzing what was there and the potential for it to cause damage and made a bad choice in terms of a rootstock choice. Oftentimes our rootstock choice is driven by availability too, and that can be a problem. Some stocks are a bit nefarious, like 1103 Paulson, which is very widely used now. It seems to thrive on almost soils and most sites for some period of time. And what's happening is it has a tremendous root system that keeps refreshing itself and keeps regenerating new growth and overcoming the pests and pressures that are in the soil. So it's useful from that perspective, I guess, as you think about it. But it slows down at some point and fails. And we're seeing this more and more in years 7 through about 10. You start seeing the effect of salt. You start seeing the effect of nematode feeding. They start slowing these plants down. The root system is declining after intense pressure, and it really makes a difference in those cases. There's a paper here that I've referenced for you that Howard Ferris and I put together quite 12 years ago now, almost, not quite 10 years ago. And it has a couple of tables in there that'll give you a good guide to all the rootstocks. There's about 30 or 40 rootstocks in there to get some idea of how they perform against different nematodes and what their backgrounds are as well. So look at that in the Journal of Entomology. So we bred new ones, the Gerin series, grape rootstocks for nematodes, the 1 through 5. They're mostly based on candycans, Champinii, Rufo-tomatosa, Riparia in their backgrounds, except for Gerin 1, it's half rotundifolia. And these have tremendous resistance to nematodes in general. So we started out by testing this group with against root knot nematode standard strains, and then against aggressive strains that fed on harmony and freedom, and then against all those strains at once with dagger nematode thrown in for good measure of Vicipium index. And then we test them against higher soil temperatures. We know that about 80 plus degrees in the soil, that the nematode resistance based on Champinii and based on most root knot nematode resistance genes breaks down and starts failing. So we tested it there as well. And then this series of stocks, these 5 stocks, rooted and were resistant against all those nematode problems. And then this series of nematodes on this slide here gives you their response to citrus, ring, lesion, and pin nematodes. So they all resist those aggressive root knots and the dagger nematode. So they're pretty amazing. And people ask me, which stock should we use for nematode resistance? And this is the answer. When they ask me a lot about its viticulture attributes, I say, well, we don't know yet. So they're fairly young stocks. They haven't been trialed in many, many sites, but they're getting more broad use across the state now. So that there's some benefit there. They were bred for Lodi initially. When I thought about where could we use these most effectively, it'd be sort of the upper part of the San Joaquin Valley, where you do have a mixture of a lot of different nematodes frequently, and that these would help answer that problem. The Gerin 1 looks pretty impressive, but again, it's hard to propagate, and it's not impossible. The nurseries are having trouble because they're not being patient enough in the calcium process. And if they would wait longer, like we do, we get 70+% rooting success, and they're getting 10, sometimes less than that. So it hasn't built up a very strong reputation, as you might imagine. But if they did switch the propagation practices a bit, they could cope with it. Gerin 5, we sort of lost interest in. It hasn't grown well in the field, and it has been consistently hard to propagate, and even with other tricks, and it hasn't moved ahead. We've always advanced that one because it was a very diverse and unusual background, species background, thinking that it'd be good to have that diversity of resistances potentially, but it's not really done very well commercially. The other ones work well. They're all sort of intermediate in terms of their vigor, and I'll have a little survey here as we go through. I don't think I'm going to talk about this one, so we don't get too far into things. How are we doing in terms of time? We're doing okay. Yeah. No,
yeah, we have about 7 minutes before the other talk starts.
Yeah, okay, good. That's what I thought. So here's the list of the Gerin parentages to give you a feeling for the diversity of these materials. We've had them in trials all over the state. The best 2 trials, one in Lodi and one in Geyserville. One with the Lodi, one was with Paul Vertigal. Now Keith Striegler's been pushing it through, which has been very helpful. And in Geyserville, Rhonda Smith's been pushing that trial through for many years as well. It's with Vino Farms. They're both very interesting sites, and the fan leaf is finally starting to express. So one of the problems with looking at these materials directly for fan leaf virus is you have to have diseased vines. And we put these vines in known infected sites, and yet it's taken years to start seeing symptoms of the virus. And we're talking 8+ years now to really start seeing them, and on the susceptible control materials. And the ones that are resistant, the question always ends up being, well, are they being fed upon, or is it the time it takes for this virus to really get well distributed and through the vine and expressive, or is it not there in the first place? And it's hard to sample for, as we've already mentioned. Some of the take-home messages: the GRN1 is very low vigor. So we keep hearing we want a devigorating rootstock. Well, here's one. GRN1, it's a very devigorating, not deathly so, but just 20-25% less vigorous than the more standard stalks in these same plots. And it has pretty high fruitfulness, which is unusual. So usually if it grows less, they fruit less. And these seem to have maintained their crop loads pretty well. And maybe that's the effect of rotundifolia and the effect on the virus. And we'll still have to wait to see what's going to happen with that over time. We've started to see the virus come in on the GRN stalks, and I expected them to, except for GRN1. We hoped it wouldn't. But we know from past experience that resistance to the nematode is not enough. You need resistance to or tolerance to that virus as well, which seems to only come from O3916 so far. We looked at RS3 and 9 in that plot, both those plots, and it's very weak, incredibly weak, which is unusual. It had done well in the Central Coast. So Larry Bettigat had some good strong growth with Mike McHenry with that stock, but it's not done well in Geyserville and not done well in Lodi. And Schwarzman is very, very weak too. So a lot of these stocks we would just discard as being too weak to grow in the first place. I think I just went through this stuff. So GRN 2, 3, and 4, they don't need any tricks to propagate. They're very straightforward and standard stocks, but they have lots of repair in the background. And they may not have enough drought tolerance over time, for instance, or other traits that may work out. And again, the GRN1 we can actually propagate pretty well if we go through there. We've made a lot of new materials that are rotundifolia based as well. 10114 by tray shed. The tray shed is a form of rotundifolia, and we've made big populations of that, and we have individuals that look very promising. This is a study where we took healthy and infected vines, so we inoculated them by bench grafting and inoculating them, and you can see in the third line down is healthy plant material on St. George. That's the most susceptible stock we had to look at. We looked at the number of berries, number of clipter, the flower parts, and the fruit set, and we get some idea what happens on a healthy plant. And the next line down shows you what happens if it's infected. You get very few berries, and you get a very poor fruit set. And when we graft onto O3916, we get the same. And there's no impact of the virus on the grapevine and on flowering and on berry set. And then we have these other selections here at the bottom, these 6 that have also done remarkably well. They're also bench grafted with infected material, and they're maintaining good yields, and they root pretty well, some of them. So those have potential promise, those 7107 population, and we'll be starting to finish the viticultural evaluation of these over the next couple of years. I will in retirement, and the new breeder will help me as well, I hope. So those will be coming along. And we need nematode, these broadly nematode-resistant rootstocks. That's going to be important over time. Multiple species, multiple types, because we're getting more and more of the mixtures rather than single species in most sites too. We need better drought tolerance. That seems to be so far to be larger roots, but we have some new projects started with some of the new faculty to look at whether we can find other forms of drought tolerance besides just vigor, just strength of the root system, whether we can find more effectively and utilize hydraulic lift, where we can pull water from deep roots to utilize it in the more shallow surface root area. We're looking at better salt tolerance, get that in there. We've been putting that in as well. And again, this virus tolerance concept will be very important too. Okay, that's what these are, and you're going to see these in trials. So we have pretty found material that looks very interesting in terms of its resistances. 4 is the highest value in these tables. It's the positive one, the resistant one, not the susceptible one. So that they're moving ahead from that perspective. And these are in field testing now on campus to look at their growth and their phenology and their effects on bloom dates and ripening dates, that sort of stuff. Here's our friend 140 Ruggieri, the best of all commercial rootstocks in terms of salt tolerance. It ranks up pretty well here. St. George does pretty remarkably well too. Schwarzman has been known by the Australians to be fairly effective for quite some time. Ramsey was used initially, but it doesn't really have any salt tolerance. Whoops, sorry. It doesn't have any salt tolerance. It just outgrows the problem, and then eventually it's more damaged over time. Here's Repair Gloire at the other end that has no tolerance of salt at all. So what are these little guys here at the end? These are things of like Acerfolia, Triglisii, Gerdiana, that are species from the Southwest from salt flat areas. And they, lo and behold, have remarkable salt tolerance. These are actually growing fairly happily in 12.5% seawater. So no, sorry, 20. It's 6%, 7% seawater, and they grow at 12% seawater as well. So it looks they look very promising, and getting both of them into the other backgrounds and maybe released directly is something that's on the platter right now too. So we've been gathering a lot of this material from all over the Southwest. And have for many years through many graduate students helping out with that process too. And now we're slowly going through this material to find out how it can be used. And it has PD resistance, salt resistance, nematode resistance, dagger nematode resistance occasionally, some more Lyme tolerance we might need over time. So lots of potential stuff for the future there as well. Okay, I'm gonna stop right there because that's the end of my talk. And I can answer some more questions about a few of these other stocks over time too. But one of the important ones is whether we can actually breed virus tolerant rootstocks, rootstocks that induce tolerance in the scion. And it looks like it's possible, which is exciting. So we've been working with FPS over the years. They've done a lot of the large scale stuff. We've been looking at it on the smaller microscopic scale, if you like, and what's happening and why does it happen. And we have material that actually reduce the symptoms based from red leaf virus issues. So it suppresses symptom development to some degree. And those are moving along at this point too. And some are at FPS and in the field testing process as well. And we have, of course, the resistance to fan leaf or tolerance to fan leaf that's found in 03916. So that's moving along as well too. Okay, so with that, thanks. And I don't know if I have time to entertain questions, but I'll stick around for the
Hey, Con,
too.
how do you want to handle there's a lot of questions in the Q&A. You're muted, Con.
Well, let's try to answer the questions as Nazareth is speaking via answering them, and then we will take them during the grower session round table live. But keep in mind that both Andy and I have a COVID test today at 1. So, all right. Well, thank you, Andy. As great as always. I want to say I will miss you, but I know you won't be going far.
You don't know where I live, though.
Yes, I do. Anyways, our next speaker is Nazareth Torres. She's one of our scholars at Oakville Station. So we have received a lot of these questions regarding effectiveness of mycorrhizae solutions in hyper-arid conditions. So she conducted this work over the course of the 2020 season, and it is already one portion of the work is already published. So I'll turn it over to Nazareth, who came to us from University of Navarra in Spain. So take it away, Nazareth. I'll mute myself.
Okay, thank you. Let me 1 sec. Okay. So thank you so much for inviting me for this session. So as Karen told you, I'm going to present this talk entitled Use of Mycorrhizae Solution in New Vineyard Plantings. As you know, growers need to face the challenge of vineyard replantation when the grapevines are not producing good berries because of red blood virus disease, trunk diseases, leaf rot diseases, and other diseases related to viruses, and also because they are producing substandard fruit quality and therefore compromising the wine quality. However, several factors need to be taken into account before replanting a new vineyard, because if we don't do it in a proper manner, it comes with a huge economic loss. For example, in Napa and Sonoma valleys, the price of replanting a vineyard per an acre is about 30 or 250, runs between 30 and $250 per an acre. So we need to make some considerations to avoid economical loss. In this context, we need to think for solutions to ensure a good establishment. So what about the soils? So mutualistic relationships between plant and microbes offer a novel approach to enhance agricultural productivity while reducing environmental costs. The plant growth promoting microorganisms could play vital roles in the maintenance of plant fitness and soil health under stress conditions. Among these beneficial microorganisms is worth noting the arbuscular mycorrhizal fungi, because they can establish mutualistic relationships with most crops and have an increasingly important role in vineyard production systems. But what are the arbuscular mycorrhizal fungi? So those fungi are a natural symbiont, soil symbiont, that penetrate the cortical cell of the roots of the 80% of the vascular plants and for arbuscules. Given their importance in ecosystems, several studies reported the effects on a wide range of plants. And as we can see on the slide, they give some ecosystem service to the plants, like increased plant resistance against biotic stresses while reducing the phytochemical input, increases the plant resistance to abiotic stresses, reduces the fertilizer requirement, improves the soil water retention, promotes the plant growth, reduce the fertilizer requirement, and improves the soil structure. So regarding grapevines, under control conditions, mycorrhizal inoculation promotes growth, drought tolerance, and nutrient uptake. And also it's known that they protect against pathogens through the stimulation of phenylpropanoid biosynthesis key genes, and also inhibit the pathogen transmission by impairing the growth of nematode vectors, their reproduction in soils. However, we don't really know if those effects are shown also under real field conditions. However, because the rootstock genotype or the type of inoculum could affect the effectiveness of the mycorrhizal symbiosis. Additionally, the mycorrhizal inoculants consist of a single or a combination of few mycorrhizal fungi, isolates grown in plant culture or greenhouse conditions with annual grasses. So they might not establish on woody plants such as grapevine, because maybe they have another different ecosystem preferences. And also, we need to take into account that establishment on the symbiosis is very dependent upon the climatic conditions and also all these abiotic factors related to climate change, as the increased radiation, increased CO2 concentration in the air or whatever. So this is what we have right now. For most wine grape producing regions, you know that they are suggested to seasonal drought, and based on the climate global models, an increase in aridity is predicted for the future. Actually, during the last decades, climate in California tended to little to no cloud cover with intermittent heat spikes, with a general warming trend, and of this without no appreciable increase in precipitation supply. Actually, for example, February of last year was the driest February since there are records. Therefore, the optimization of the irrigation schedule would still be one of the most desirable tools to improve crop productivity and quality. Actually, in many historically non-irrigated areas, irrigation is expanded fast to overcome these environmental stresses. Sorry. And also in other areas like California, that is warmer and drier, these areas rely on irrigation for crop production, and water resources are becoming scarce. So we need also the optimization of this irrigation schedule. So what's next? Today, wine growers are aware of the importance of sustainable viticulture that ensures the profitability in the future without compromising berry quality. However, little is known about the contribution of that the mycorrhizal fungi can have for implementing the effect of different irrigation amounts on the performance and berry quality of young grapevines and the real field conditions. So our aim in this study was to characterize the response of young Merlot grapevines to mycorrhizal inoculation subjected to two different irrigation amounts in their first productive year. So to achieve this aim, we conducted an experiment at the Oakville Research Station from the UC Davis. The vineyard was planted to Merlot, grafted onto 3309C rootstock at a space of 6 by 9 feet row per vine and oriented in east-west. Plants were spread to a quadrilateral trellis system at a height of 66 inches from the floor, vineyard floor, and they were drip irrigated once a week with 1 or 2 emitters, depending on the irrigation amount, that they have the capacity of deliver 1 gallon per vine and per hour. And the meters were spaced about 6 feet in the dripping line, sorry. So the experimental design was the following. You can see here our vineyard and how the different treatments were placed in the vineyard. So we made that 2x2 factorial design in a split plot, and with different combination of non-inoculated or inoculated grapevines with the arbuscular mycorrhizal fungi, irrigated with full irrigation treatment, or with the half amount of water that we call half irrigated treatment. So here you can see the mycorrhizal inoculum that we used was called MycoApply Endomax. It's a commercial one that you can get easily, and it's composed by the different propagules of Rhizopagus intraradices, Funeliformis mose, Glomus aggregatum, and Glomus etunicatum, that are the main arbuscular mycorrhizae found in many areas. The inocula has more than 5,000 propagules per gram, and it was inoculated at a range of 10 grams of inoculum per 1,000 plants. And the application took place on March. Then the irrigation treatments were done based on different fractions of the crop evapotranspiration that was calculated as the equation shows, as the product between the ETO, that is the evapotranspiration, and the crop coefficient, Kc. So plants, fully irrigated plants receive the 100% of the crop evapotranspiration, while the half irrigated plants receive the half of that amount. And the application of the schedule of the irrigation was applied weekly from the beginning of the summer, about in May. We started with the irrigation, and then we finished it at harvest. So during the experiment, we monitor several variables related to the grapevine water status and gas exchange parameters, the canopy architecture, yield, components, and mineral content. And also we made some chemical analysis on berry to check the berry composition and the primary and secondary metabolite. So what we got? Here we can see, we are going to see the results. So the climatic conditions of this 2020 growing season were quite weird as the whole year. So the comparison of the growing season of the experiment and the reference data for the same period within the last 30 years, so that the 2020 was warmer and drier, so that the average daily temperature was about 33 degrees Fahrenheit higher, reaching more than 35 degrees more in August. And precipitation was about 530 millimeters less. I think that is something like 31 inches less of precipitation supply. So 2020 growing season seems to be a strange year regarding temperature and rainfall. Well, the second step was to know, are these mycorrhizal fungi improving the grapevine growth? So we monitor first the mycorrhizal colonization intensity. I'm going to explain briefly how we did it. So we sampled the roots, cleaned them and stained, and then analyzed them under the microscope. So you can see in the second and the third picture the fungal structures, the arbuscles, and also the hyphae and the vesicles of the fungi in symbiosis. So we conducted this analysis before treatment application to know how was our vineyard before the treatments, and the native mycorrhizal colonization didn't show differences between the different blocks. So then we analyzed this mycorrhizal colonization intensity after 3 months and at harvest. And we saw that the blocks that were inoculated with the mycorrhizal fungi were increased the mycorrhizal colonization rates, reaching like about values threefold higher than the non-mycorrhizal ones. Then I want to explain briefly this index that stands for Relative Mycorrhizal Dependency Index. And it's a parameter that allows us to assess the dependency of a crop of the mycorrhizal symbiosis for reaching their maximum growth for a given condition, environmental condition. So we can see here that the plants that were half irrigated were really dependent upon the mycorrhizal condition to reach their maximum growth. What about the growth of the vine? So we check the vegetative growth, and the only effect was due to the half irrigation, and where plants subjected to this half irrigation treatment decrease the green pruning, the trunk diameter, the leaf area, and also the yield. However, we didn't find any difference because of the treatments in the leaf area to free radio. So these results suggest that the treatments didn't affect the balance between the vegetative and reproductive growth in grapevines. And also we didn't find any difference regarding the mycorrhizal inoculation or not. Then we check also the leaf mineral content of the grapevines, and we saw we didn't find any difference in the main macro micronutrients found in the leaf blades of these grapevines. So what's next? We wanted to know if the mycorrhizal inoculation improved the plant water status and the leaf gas exchange parameters. So we monitor the grapevine water status as the stem water potential values during the whole season. And we saw that the half irrigation was the main factor affecting the stem water potential at noon of these grapevines. However, before variation, we saw that the mycorrhizal inoculation improved the water status of these grapevines, especially when they were suggested to also to have irrigation. Then we wanted to know if those treatment affected the photosynthetic capacity of these grapevines. And we saw that the carbon assimilation rate was decreased by the half irrigation treatment, but also was increased by the inoculation of grapevines when they were suggested to the full irrigation. Regarding the water use efficiency, instantaneous water use efficiency, the inoculation of grapevines improved it in combination with the irrigation treatment. And then the stomatal conductance was highly affected by the irrigation treatments, where half irrigated plants decreased these values. However, we saw that the mycorrhizal inoculation of these plants improved the stomatal conductance. So finally, we wanted to know if all these changes in plant physiology affected also the berry chemistry and composition, and consequently the berry quality. So we didn't find any difference on the primary metabolites that we measured. So no difference on the mass pH, acidity, or total soluble solids. However, we saw that the both treatments interact on the very fresh mass. Therefore, the inoculation of grapevines under half irrigating conditions decreased the very fresh mass, while the inoculation of these grapevines under fully irrigated conditions increased the very fresh mass. Regarding the secondary metabolites, we were focused on the flavonoids. So as you know, flavonoids are important molecules for berry quality due to they contribute to color stabilization and also to the antioxidant properties of the wines. And we saw that the irrigation treatment was a main factor affecting them, decreasing the derivatives of quercetin, lecithins, camphorols, isoarmetins, and silichitins. However, we didn't measure any difference in the total amount of flavonoids, but we saw that the mycorrhizal inoculation under half irrigated conditions improved some quercetin derivatives. On the other hand, we check also the anthocyanin composition, given the importance of anthocyanin for the wine color and berry quality. So the most affected derivative was the cyanidin, which was decreased under half irrigated conditions. But we wanted to know if the arbuscular mycorrhizal inoculation has something to do with this, with the flavonoid metabolism. metabolites. So we check the relationship between the rates of the mycorrhizal colonization and the flavonoid contents, and we saw a strong positive relationship of the mycorrhizal colonization with the total cyanidins, pionidins, and quercetins found in merlot berries. So to finish, we are going to discuss a little about the profitability of these treatments and the sustainability of them. So we are going to see the results of flower cost as well as the water footprint of these treatments. So we saw that the mycorrhizal inoculation had almost no cost for the growers. However, irrigated vines with half of the amount of irrigation decreased the total cost, but also decreased the yield. However, these differences were not strong enough to decrease the gross income that the growers receive for the berries. The water footprint, as you know, it has three components. It is composed by the green water footprint, or the amount of water that comes from the rain, from the precipitation, the blue water footprint, or water source from irrigation, and the gray water footprint, that is the amount of water that we need to dilute the pollution to reach some water standard quality. In our trail, we didn't check the gray water footprint because we didn't put any fertilizer or herbicide in the blocks, just to see the effect of the mycorrhiza. So our water footprint is the same between the green and the blue water footprint. And we saw that the half irrigation treatment was the main factor affecting them, increasing the green water footprint and decreasing the blue water footprint, would that lead to a decreased total water footprint. So in the first productive year of this merlot grapevine, we saw that the total water footprint could maybe decrease by the half irrigation treatment. So I want to address these conclusions from this talk. So we saw that the mycorrhiza inoculation improved vegetative growth, photosynthetic activity, water status of grapevines grown in fields, especially when they were facing mild water stress. Also, the symbiosis between grapevines and mycorrhizae regulates the anthocyanin and flavonoid metabolisms. And the mycorrhiza inoculation was not sufficient to avoid the yield losses due to the half irrigation treatment, in spite of the benefits that we saw in some berry traits. In this first productive year of merlot grapevines, when they were facing an hyper-arid growing season. So we can say that this result may be affected by the edaphoclimatic characteristics and the living microbiota in the vineyard soil, which should be taken into account before making the decision of inoculating or change the irrigation management. So to finish, I want to thank Doctors Can Cultural and Rusio, who helped me to conduct this experiment, and also the funding that we received for doing it. And if you want to read more about this study, you have here the reference of the study is published. It's already published and it's open source, so it's available for everyone. And if you have any question, I will be happy to answer by mail or in the chat or whatever. Thank you.
Okay. Well, thank you, Nazareth, for a great talk.
Okay.
So I lost my internet connection for a while, but now I'm back.
You know the study.
Yeah, I know the study. So anyways, so we do have some time for questions for Nazareth, then I will open it up for a greater discussion in a couple of minutes. Christina is on another call, so she will join us after a while
Okay.
for those. So Nazareth, you have some questions from the growers. The first question is it necessary to reapply AMF every so often, or once you apply, they stay?
Well, this is a really good question, and this is the second step that we are going to do, because there are few studies in field. So when you check the effectiveness of mycorrhizal inoculation under greenhouse conditions, it's just 1 year. So the results, you cannot translate these results into field conditions. So there are some papers, but very few, that they were studying this effect, like reinoculation. So there is lack of knowledge about that. If the improvement that we can get from the symbiosis is maintained in the time. I cannot give you a more focused question answer. Sorry.
Another question is, are there any plans to perform a similar experiment on mature established vines, ages 5+? Also, how long lasting are the increased levels of AMF on the inoculated grapevines?
So the inoculation reaches values of 3-4 higher. The colonization rate were 3-4 higher. But the thing is that, as Christina told us before, one thing is the structure and another thing is the functionality of the microbes. So what we saw is that we have more mycorrhizal symbiosis, more fungal structures, but we don't know if those structures are effective or not, or are functional, in fact. So there is another, that is another question that we need to assess in the future.
Yeah, true. So a question from Bob is it possible that moist soils already have a mycorrhiza in the soil or on the grape roots at the planting time, so inoculation is unnecessary?
Yeah. Well, with this study, because there was an idea that if you inoculate, the fields are, I mean, the vineyard soils have a mycorrhizae. They have this species naturally. So there was a whole idea that if you inoculate the field, the new mycorrhizae remove the natural ones. But what we saw with this experiment was that it's complementary, at least in colonization rates. So now we are looking into the soils and also how this mycorrhizal inoculation affect the bacterial and fungi communities in these soils. So we need to study more about that, about if the effect of colonization it's beneficial, or it's or you cannot get anything else from the inoculation of fields. But as I know, I think that we need to continue study this, because all the studies are in greenhouse conditions. So we cannot answer that question today, and growers are confusing about using them or not.
Yeah. Well, one thing you might want to mention, Nazareth, is that when we ran the applied water mouse study, the ET crop fractions study, you were able to change the mycorrhizal colonization in these vineyard soils. You might want to mention that.
Yeah, of course. Yeah, sorry, I didn't answer the first question. The moisture of the soils, the mycorrhizal, at least as I say, the mycorrhizal abundance on the soils is very dependent upon the soil moisture. So we check also in another experiment that wasn't inoculated just with the natural mycorrhizal found in the vineyard, and we reported that the mycorrhizal colonization was increased with the decreased water amount that vine received. So we found a negative relationship between the soil moisture and the colonization rates.
So the question is, next question is a big one. Have you measured the interaction between mycorrhizal and endophyte fungi working together? So this is the interaction question for the microbiome, what you're working on now.
Yeah. I'm working on that. So yeah, at some point it affects, but we didn't find like a huge effect on the functionality of these microbes with the treatments. So we don't know if it's because it's the first year, we need to reinoculate. So I'm sorry not to give you like a real answer, but this is what we know about this at this moment.
I mean, there's more questions than answers we have at the moment regarding microbiomes of vineyard soils, I think. So it's just scratching the surface. So Guillermo from Davis asks, is there a systemic fungicide that should be avoided in order not to affect the mycorrhizae?
Sorry, can you repeat, because the internet was...
Is there a fungicide that's applied for plant protection that should be avoided not to affect the mycorrhizae?
Okay. I guess that is depending on, I'm not an expert on fungicide, so, but I guess that is depending on the selectivity of the fungicide.
Yeah, I mean, this vineyard was sprayed about every 11 days, and we didn't see an adverse effect of the systemic ones that we used.
No.
Question is, did you cultivate or are you planning to? Meaning that did you till the soils or are you planning to till these soils where this work was done?
At this moment, my first idea was not to touch the soils. So because I wanted to see to make the experiment as the more control that we could under these real conditions. So the soil was untilled, and we have the natural grower there. And also we didn't put herbicide or fertilizer on these rows.
Yeah. Yeah, so these rows were managed like we normally would manage, meaning that they were not tilled, and mustard was allowed to grow in between rows, which is kind of like what comes up in these oak-ville soils. So another question is, are there any resources you would recommend for learning more about the different types of microbes, particularly the ones in mycorrhizae inoculations?
Any resources?
Like
so
books, papers.
Yeah, I think that unfortunately for the big audience, we have just the commercial inoculum information. So we have that and some papers. So I think that we need to join these two source of information, because sometimes, okay, the commercial people, they are going to say, oh yeah, my inoculant is the best, you are going to get this, this, and this, and then you inoculate your field and you don't have anything. So I think that we need to study more and also to make this bridge between the papers, the scientific community, and the growers.
I agree. Another question is there any risk of introducing these non-native mycorrhiza, meaning that could they shift the soil microbial populations in a negative way?
At some point, yeah, this is like in next chapter.
There's always a next chapter in research.
Yeah, at some point, maybe. But what we are seeing today is like the fruit quality and the grapevine performance was improved after inoculation, or at least wasn't like impaired. So we can say that it's good. It's okay. We didn't see like a huge effect or a huge benefit of inoculation, but we didn't see neither like a detrimental effect of it. So on the microbial communities, yeah, some of the microbes that the inoculation enhance, they are pathogens. But it's true that the microbial community is a net between fungi, bacteria, archaea, and all this stuff that they are collaborating, and the functionality of them it's made by all of them. So we cannot say, oh, it's improving this pathogen. Yeah, but maybe it's improving also another one that is the opposite, and the function are like delete or something like that. So we didn't see any detrimental effect, and what we saw was fine. Was okay, an improvement in the flavonoid metabolism. So
No.
There's a couple of questions here for Andy Walker. If you're still listening,
I'm here.
able to answer. I know you're typing answers to some of the questions, but
Yep,
there's
yep.
a question about whether you can touch on RS3, RS6.
I haven't got to that one yet. They've done very strangely, very, very poorly in the two sites that we're most confident in terms of their growth and establishment and what's there. Yet they've done pretty well on other sites. So I don't know what's going on in terms of from the data we've seen, or the plant growth we've seen, the data we've collected of these two sites. I would never use those rootstocks anywhere. Yet we've seen pretty good development in other spots. So I don't know if it doesn't seem to be a soil specific issue. One is a pretty typical loamy soil in Lodi, and the other one is a more gravelly, difficult soil in Geyserville, where you'd expect vines to grow more poorly, but they're doing poorly in both spots. It's unusually poorly. I mean, it's just weird.
Yeah, we removed them from our evaluations at Lake County and Oakville, the RS series.
Yep.
Because we planted 10 reps, 9 vine panels of each, and we were left with like 2 or 3 at the end of first year. So we removed them.
Yeah, it's peculiar because it really did do pretty well initially.
And there's one more here for you, Andy. Has there been any have there been any breeding efforts on selecting for traits with natural toxicity towards plant parasitic? Actually, this one might not be for you. This is nematodes and common cover crops. So this one actually may be for previous.
You could inoculate them. That was for you, inoculate the cover crop with arbuscular nematocidal compounds. I don't know. No, the short answer is no. There's been a lot of work on looking at toxicity of compost made from different weed species and different plant species that have some natural nematocidal activities. But in general, you need so much of this stuff in a solid mass that it becomes impractical. and it doesn't affect things very deeply in the soil. So grapes have deep roots, and most of these studies have been done on annual plants where they have very shallow root systems, and you can demonstrate an impact, but it'd be hard to really do anything on grape. It may help suppress things. That the most active level, soil out level is down, 6 to 12 inches in the soil, but I don't know. In general, no, I guess. No work that's been done from a breeding perspective.
And there's one more here that mentioned some of Khan's presentation from yesterday. He mentioned very plastic clay soils can induce water stress on vines. Would you recommend a more drought tolerant rootstock like 110R or 1103P on heavy clay soils instead of 161 or 1616C?
Is Rhonda still there? Rhonda and I looked at a plot years ago. She was running a rootstock trial on a mucky, what was supposed to be a saline site in the southern Caneros region and more on the Sonoma side of things. And all the rootstocks did really poorly, except for 1616C, which thrived. That mucky site. And now looking back, maybe it wasn't 1616C. That's one of the possibilities. It was always in the mix there. I don't know. I guess the safer answer. Not enough work has been done to really make that conclusion.
And then
I think 110 really is drought tolerant. 1103 is not, probably. It survives drought by just producing new roots wherever it can, whenever it can. And it's not going to do as well in a real dry situation, truly dry situation, where 110 over time will dig down deeper and probably be more stable. It doesn't have a strong start, but has a good strong middle and good strong finish in terms of its productivity and development with less water. You could get equivalent growth and development with less water on 110 than on 1103, I think.
Now, this is for any presenter. Someone asked, are there any beneficial fungi and or nematodes that may help reduce nematode pests? Anyone want to pick that one?
So if you want to easily find that stuff out, go look through Mike McHenry's work. He's done quite a bit of that work. And I think Andreas is also Westphal is picking up on some of it too, where they did a lot of studies in tanks to look at those sort of issues and effect on nematodes. In general, you're better off with resistant rootstocks. And the unfortunate part is that means you have to change what you've done, pull them out, start again, or just think about it more carefully the next replant.
This might be one for Nazareth. Is there some difference to inoculate roots before planting by immersion and by irrigation system after planting?
Well, sorry. I don't think that there is any difference depending on the formulation of the inoculum. Some of them are made for doing before planting, and another one is with the irrigation. The establishment of the mycorrhiza can be achieved with both of them. So I work in the past with both kind of inoculation, and we have mycorrhizal structure in both of them results. So I don't know. I think that is depending on the combination of the isolates, and if they are and if the grapevine can establish mycorrhizal symbiosis with them.
This is kind of an interesting one that someone asked. Is horse manure compost known as a good compost in vineyards? Anybody want to take up that one?
Probably depends on the feed.
Yeah.
How salty the grain is and everything else too.
Yeah, because they add salts to a lot of these feeds, and that might not be as desirable with the commercial rootstocks we have, unless you have like the Ruggieris and other stuff. So, I mean, we have a lot more unknowns than we have knowns when it comes to like managing soils and what's underground.
And someone was asking, I think this is for you, Nazareth, how does your work compare to Kendra's work?
What was that?
That was Khan. I don't know what that was.
I'm sorry, but I'm not familiar with that. So
Okay.
We cited her in the papers. Kendra Baumgartner from USDA.
Baumgartner. Sorry, I don't know the people, and I know the
The citations.
surname. Sorry. It's quite similar. The thing is that we are studying the effect of this mycorrhizal also with irrigation. And as far as I remember, they were doing also tries with irrigation and mycorrhizal, but I guess that they didn't apply mycorrhizal inoculum onto the fields, if I'm not wrong. And also as I said, the daphoclimatic condition of the soils make the difference. So we need to do this. Actually, for example, as Christina told us before, the microbiome analysis, they are super different in a very short space in the vineyard. So I guess that the effect of the mycorrhizal may be the same. I don't know if I answered the question or sorry.
Here's actually a question for any of the panelists. This is interesting. Do any of the panelists know of plans from grape or wine brokers to offer monetary incentives for carbon sequestration in soils through the use of cover crops or compost?
CDFA is working on monetizing the carbon sequestered by soils, by agricultural lands. However, as Christina showed, there's not a whole lot of opportunity to sequester carbon in soils permanently, because it's in the first 5 to 10 centimeters of the soil. So that remains to be seen, and there are no incentives for it at the moment, because there's no legislative framework right now in California, but it is coming. There's a non-governmental organization that has taken this on and has been working with CDFA and the state legislature to monetize this. And right now the state is not allowing for sensing carbon sequestration. It is still going to be, pulling soil cores, sending it to a lab, and then getting results back. So it's going to be a few years before this is monetized.
All right. You see any other questions, Khan, that need answering?
There's one. How about foliar applications of oligoalginates and oligochitosands affecting mycorrhizal effect and time?
Okay, I was reading that question. I don't know just that molecules. I know that other molecules, when they are applied on as, for example, there is interesting research conducted on grapevines also with foliar application of potassium, but I don't know with those molecules. And of course, the effect in this study that I mentioned, the effect of mineral uptake from the mycorrhizal vines was highly affected by the phosphorus application of on the leaves. So I guess that with these molecules we will have, but I need to check the bibliography. If you give me some moment, I can give you some research about that.
All right.
There is one here. Do you have any experience using fungi to support rootstock in nematode control like Arthrobotrys oligospora?
No. That's an easy one. But it shouldn't be so flippant. I guess it's possible. So, you know, a lot of the decay you see in root systems is not physical damage from the nematodes and or phylloxera. It's secondary. It's a crack or a wound and injury that's allowing the entry of zoonosis fungi. And we found with, at least with phylloxera, that organic soils that were heavily treated, composts and other situations were there were less frequent phylloxera in those soils, and they were less damaging for more years. And in the end, they still killed the vine, but it probably delayed that process by years.
Here's another one for you, Andy, specifically. Why does Sauvignon Blanc not seem to care about the red blotch associated virus? Is this common with Sauvignon Blanc?
That's a good question. And we don't know that, but there will be variety specific responses. And tolerance is probably like we see in other virus situations too. And there's probably there might be a route to helping to solve some of the problems too, if you knew that better.
Yeah.
Sauvignon Blanc does not show symptoms, but carbon translocation is certainly affected under red blotch infection. So the interesting thing about what we're finding about red blotch is that white grapes are harvested early. So that's why growers seem to think that it is not affected, but it is, you know, affected just as much as a red wine grape. Chardonnay, Sauvignon Blanc, all these are, you know, affected. They just do not make the anthocyanin in the leaves like reds do. So if you were to wait till 26, 27 bricks, you'll see the same thing.
Different response.
Different
Raisin
response
Blanc.
from Sauvignon Blanc and Raisin Blanc. So.
Great.
All right, I think we're good for now. Oh, here's a question, Andy. Did own-rooted Thompson seedless have some natural tolerance to nematodes? Old raisin vineyards in the valley just keep cranking out 2 tons in super sandy soils.
Yep, probably 20 tons, I hope. Or 2 tons of raisins, maybe.
Yeah, 2 tons of raisins, yeah.
So yes, and has to do with that rooting system and the fact that when they planted those vines, the soils weren't as badly infested or not at all infested. And nematodes have come in afterwards, but they're not able to have as big an impact on that massive root system. I mean, the vinifera root system is really impressive, and the table grape types are even more impressive than the wine grape types.
Yeah,
they're deeper, they're more intense, they're more uniform in their structure from top to bottom in terms of the density of those roots. They just outgrow them. They're able to survive. But if you replant that same site with fresh plants, you can forget it most of the time.
Are you planting a Thompson vineyard? Come on.
Me? No, I don't think so.
Only my dad replanted one at the age of 72, and I was just like shaking my head. Anyways.
There's one that you were, I think,
answering.
Is there any management under the vines that could help reduce parasitic nematode damage? And that, I think, was going, Andy, you were typing an answer maybe.
Yeah, I think organic practice, again, helps reduce the damage, not the numbers necessarily, but the damage, because it's the interaction with the beneficial and pathogenic or at least saprophytic fungi that are causing a lot of the damage. But it's not a cure-all for sure.
No, I think. Okay, I think we exhausted all the questions for today. So thank you again to all the panelists. Great talks as always. You will receive the email later this afternoon with the recordings, the PDFs of the talks, and your CE credit questionnaires and quizzes and all the pertinent information. So watch out for that. So I'd like to thank Caroline and Karen for putting everything together. Of course, all the panelists and all the funding we received to do soil health work within the last year from American Vineyard Foundation and Napa Valley growers, and of course all the other work that is being funded by our wonderful community. So I think with that, I'd like to wish you guys a good afternoon, and then we will see you in the next program, which is March 4th, winery regulations.
Winery regulations, that is correct. Be on the lookout for the registration.
Be on the lookout. All right, see ya, stay healthy, get COVID tested, and then we will see you soon. Bye-bye.
Bye-bye.